With your permission, can I start, sir?
Neha, you can start now.
Thank you, sir.
Nilesh ji.
Very good morning to all the distinguished dignitaries, speaker delegates, and participants. I, Neha Kamalpuria, Professor, Indore Institute of Pharmacy, extend my heartiest welcome on the second day of scientific session National E-Conference on Translational Research for Nanomedicines, organized by IIP, sponsored by AICTE in association with IPGA. We are happy to inform that on the second day of virtual national conference, we have scientific session by Dr. Sanyog Jain, Professor, Center for Pharmaceutical Nanotechnology, Department of Pharmaceutics, Naipur, Mohali, on the topic Design of Cancer Nanomedicine with Improved Therapeutic Efficiency and Safety with Chairperson Dr. Manish Chaurasia, PhD Senior Principal Scientist, Pharmaceutics and Pharmacokinetic Division, Central Drug Research Institute, Lucknow. And second scientific session was taken by Dr. Rakesh Kumar Tekade, Associate Professor, Naipur, Ahmedabad, on the topic Prospectus and Retrospectus in Nanomedicine, with Chairperson Dr. Shashank Kumar Singh, Senior Principal Scientist in Pharmacology Division at CSIR, Indian Institute of Integrative Medicine, Jammu. Scientific session is followed by poster presentation. For poster presentation, our chairperson is Dr. G.N. Dharwekar, Principal, Acropolis Institute of Pharmaceutical Education and Research, Indore, with the co-chairperson, Dr. Preetam Siraskar, Manager Production at Glenmark, Chhindwara. Then after lunch, we have validatory program with our chief guest, Mr. Amresh Gautam, Head Product and Manufacturing, Zydus. Now, with due permission of our eminent speaker and chairperson, I would like to proceed the technical session. Sir, may I proceed? Can we start our technical session?
Neha, just wait for 2 minutes. I'm.
Yes, sir.
Yes.
Hey,
next previous करना. Yes, Neha, you can proceed.
Thank you, sir. So our first scientific session was taken by Dr. Sanyog Jain, Professor, Center for Pharmaceutical Nanotechnology, Department of Pharmaceutics, Naipur, Mohali, on the topic Design of Cancer Nanomedicine with Improved Therapeutic Efficacy and Safety, with Chairperson Dr. Manish Kumar Chaurasia, PhD Senior Principal Scientist, Pharmaceutics and Pharmacokinetic Division, CSIR, Lucknow. Sir is awarded by CDRI Annual Research Incentive Award for research publication in journal having impact factor more than 7. Sir is awarded for CRS capsule Pfizer innovative aspect of oral drug delivery and absorption graduate postdoc award, Honolulu, Hawaii, USA. Sir is awarded GP Nair Award by Indian Drug Manufacturers Association, Mumbai, India. Sir also has received grant for several projects by various government organizations worth 2 crore 66 lakh. Sir has 70 publications, 12 book chapters, and also guided 10 PhD scholars. Now I request Manish sir to introduce our eminent speaker, Dr. Sanyog Jain. Over to you, sir.
Can you hear me?
Yes, sir, you're audible.
Okay, I'll take my privilege to introduce our eminent speaker, Professor Sanyog Jain. Currently, he is working as a professor at Center for Pharmaceutical Nanotechnology, Department of Pharmaceutics of National Institute of Pharmaceutical Education and Research. Previously, he has worked as scientist at a Defense Research and Development Organization, that is DRDO, Ministry of Defense and Government of India. Dr. Jain's key research interest includes using different types of nano drug delivery systems for various biomedical applications, including targeted anti-cancer drug delivery, oral delivery of vaccines, proteins and peptides, and other therapeutics, co-encapsulation of high-dose drugs, especially in antioxidants, topical and transdermal drug delivery, gene delivery, and carbon nanotubes. Dr. Sanyog has patented several nanotechnology-based drug delivery platforms and licensed tamoxifen SEDDS technology for improved breast cancer therapy to industry for commercialization. He is inventor of 16 patents, out of which 11 patents are already granted. He has authored 2 books, 11 book chapters, and published more than 200 research papers in high-impact international journals with more than 10,000 citations. Dr. Sanyog has won several prestigious awards, including AAICTE's Distinguished Young Educator and Research Scholarship Award, ABAP Gold Medal for Advanced Research in Nanoscience and Technology, Shivnath Rai Kohli Memorial Mid-Career Best Scientist Award, Pharma Innova Award for Best Research Guide, Bharat Jyoti Award, Rising Suns in Asia Award, Alkyl Amine Young Scientist Award, INSA Medal for Young Scientist, OPPI Young Scientist Award, and Punjab State Young Scientist Award for his outstanding contribution in the field of nanomedicine and drug delivery systems. He also secured rank in list of world's top 2% scientists in the field of pharmacy and pharmacology, which is published by Stanford University and Elsevier in 2022 and 2021. He has supervised 2 postdocs, 14 PhDs, and more than 100 master's level projects, and currently supervising 1 postdoc, 10 PhDs, and 10 master projects. Dr. Jain is a widely traveled and recognized scientist. He has delivered over 160 invited talks across the globe at numerous INR grants by various funding agencies and pharmaceutical industries. Dr. Jain is serving as expert committee member of Indian Pharmacopoeia and Radio Pharmaceutical Committee of DAE Government of India. He is an editor of editorial advisory board member of various international journals like Molecular Pharmaceutics, APS Pharm Science Tech, Drug Delivery, Translational Research, and Biomed Research International. And so I summarize that he has done an excellent job in the field of drug delivery systems and an eminent personality throughout the globe. So I request Dr. Jain to deliver his talk.
Thank you, sir. I request Dr. Sanyog Jain, sir, to proceed the scientific session. Over to you, sir.
So a very good morning to everyone. I hope my screen is visible.
Yes, sir, it is visible.
Yeah. So thank you, Manish sir, for very kind introduction. Thank you, Dinesh, for kind invitation to share some of my experience in the field of cancer nanomedicine with all of you. So I will not waste much time on discussing the cancer statistics. I will only say cancer is still a major cause of death worldwide. Okay, it is a global problem. It is an unmet challenge itself. Okay, or current cancer therapy involves like surgical removal of the cancer, conventional chemotherapy, radiotherapy, and hormone or adjuvant therapy. Okay, so these therapies are available. So my talk, okay, related to cancer nanomedicine will focus on co-administration of two drugs for synergistic therapy, targeted intravenous therapy, that is using multifunctional nanocarrier, oral cancer chemotherapy, and localized transdermal delivery for superficial cancer like breast cancer, okay, or skin cancer. Okay, so this figure shows the drawbacks associated with the conventional chemotherapy. So conventional chemotherapy is fraught with several challenges. Okay, the deleterious side effects, that is because of the non-specific biodistribution or tissue distribution of the drug. So drug which we administered, so it is taken by the non-specific or normal tissues, and the anti-cancer drug invariably kill the normal or healthy cells as well. So this is the major problem. And due to repeated exposure of the cytotoxic drug, so this cancer cell, they also overexpress the efflux transporter, so that lead to development of the multi drug resistance problem. Okay, so co-administration of the two drug combined therapy with two or more drugs, it has been emerged as a promising strategy to suppress the cancer drug resistance, and it also exhibit the synergistic anti-cancer response or therapeutic effect, because the two different drugs, they act on different stages of the cell growth cycle. Okay, so like one drug, okay, it acts on the G phase, another drug which acts on the S phase. Okay, so it is possible if we treat the cancer with just one cell, so the cancer cells which are on the particular growth stage cycle, say S phase, okay, they will only be killed, but the cell which are not in the S phase, or they already crossed the S phase, okay, so they will remain survive, and ultimately they will become resistant to the drug. Okay, so if we have a combination therapy with two drugs, so it will lead to overcoming the problem of this drug resistance, as well as it will lead to synergistic anti-cancer response. Okay, but there are also several limitations of the conventional combination therapy. Okay, like in the conventional combination therapy, we administer two drugs simultaneously, or one drug after another. Okay, so because the two drugs may have different physicochemical properties, like different solubility profile, different permeability profile, and their ADME profile would also be different. Okay, so it is not possible to have the simultaneous availability at the cancer site, okay, at the same time. Okay, so unifying the pharmacokinetic, it is a cellular uptake of the two drugs at the cancer site, it is a challenging task. Okay, so this problem can be overcome if we try to encapsulate the two drugs in the nanoparticle formulation. But again, encapsulation of the two drugs with a different solubility profile or different physicochemical profile in the same nanoparticle with same encapsulation efficiency and the same release profile, it would be extremely challenging. Okay, so in this regard, this combinatorial conjugation of the anti-cancer drug, okay, it holds tremendous potential to synergize the cancer chemotherapy. So if we conjugate the two drugs, okay, with the help of some spacer, so that will lead to like a hydrophilic hydrophobic balance or HRB balance, like solubility, optimum solubility we will get. And it can also overcome the drawbacks associated with the conventional therapy because of the multi-target mechanism, and it will lead to synergistic anti-cancer therapy. And because the two drug molecule, okay, would be in the same bioconjugate, so we can also achieve the unified pharmacokinetic and cell uptake. Okay, so this is the hypothesis. Two drug molecule with opposite solubility profile, they can be conjugated together with the help of some spacer like polyethylene glycol. So gemcitabine, it is a extremely hydrophilic drug. Okay, it can be conjugated with curcumin or docetaxel, which are practically insoluble drug. Okay, so this combinatorial drug conjugate, Okay, so it will have optimum solubility profile. Okay, and because of this PEG chain, so it will also show a prolonged circulation half-life. It will also like it will be resistant to the metabolic degradation. So gemcitabine is the very susceptible to metabolic degradation. I will explain in more detail in the next slide. Okay, so ultimately we will have this improved pharmacokinetic and improved anti-cancer response, or ultimately we will get synergistic anti-cancer therapy. So instead of having this two drug molecule, we can have one anti-cancer drug and one antioxidant like curcumin or quercetin also we can take. Okay, or beside that we can take one anti-cancer drug and one endogenous targeting ligand. Okay, so to make this bioconjugate self-targeted system, okay, like we can conjugate with HER2 antibody. Okay, so that can be a self-targeted system for the breast cancer therapy. Okay, so we took gemcitabine and docetaxel for our study. Okay, so as I said earlier, okay, gemcitabine, it is a very hydrophilic drug, extremely hydrophilic drug, and its half-life, it is very less, around 30 to 90 minutes. Okay, so the problem with gemcitabine, it undergoes rapid metabolism in the blood circulation. So gemcitabine, it is a difluorodeoxycytidine analog, so which is converted into the difluorodeoxyuridine analog, DFDU. Okay, which is inactive in nature. Okay, very rapidly, and because of its high water solubility, its permeability is less, so its cellular uptake is less. So that's why we need to administer it in conventional form. Gemcitabine dose is very high. Okay, on the other hand, this docetaxel, it is practically insoluble drug, and the conventional formulation of the docetaxel, that is Taxol, so it contains 280 as a surfactant and ethyl alcohol, okay, up to 40% concentration to solubilize the drug. Okay, so not only the toxicity is because of the drug, but toxicity is also because of the this 280 surfactant as well as ethyl alcohol, organic solvent. Okay, and in the conventional form, of course, because the two drugs are having totally opposite solubility profile, so their PK profile, okay, would be different. Their cellular uptake is also different. And if we try to encapsulate in the same nano carrier, so poor drug loading, it is observed. Okay, of course, if we try to encapsulate gemcitabine using hydrophilic carrier system, so the loading of docetaxel will go down. If we try to encapsulate docetaxel, then gemcitabine loading will go down. And this combination, okay, so as I explained earlier, because two drugs, they act on different mechanisms. So docetaxel, it is microtubule inhibitor, and this gemcitabine, it is an anti-metabolite drug. So both drugs, they have different mechanism of action, so that's why they will lead to synergistic anti-cancer therapy. And very important fact is both it shows like a hematological toxicity, while this docetaxel, it mainly lead to this hepatotoxicity or nephrotoxicity. Okay, so non-overlapping toxicity is also important. Okay, if we combine the two drugs, then they will not precipitate any particular toxicity. And in the conventional form, this combination, it is approved very well. Okay, so this direct conjugation or this short chain linker, they were not sufficient, means they did not offer the sufficient flexibility for the molecule to interact with the receptor. Okay, and this higher chain length PG, it increase the solubility. Solubility majorly predominated because of the PG, so it also inhibit the PG, and because of the steric hindrance, so it limit the cellular uptake. Okay, so we found this PG 2000 was optimum, so we further proceeded with PG 2000. Okay, so it resulted in this amphiphilic dual drug conjugate. Okay, gemcitabine and PG. Okay, that is hydrophilic in nature, and docetaxel, it is hydrophobic in nature. Okay, with optimum solubility and cell uptake profile. And because this is a amphiphile, so like other amphiphile, it also exhibit the property of self-assembling. Like any amphiphile, if we disperse in aqueous media above their CMC value, so they self-assembled in the form of micellar structure. So this amphiphile dual drug conjugate, it also self-assembled in this self-assembled nanoparticle. Okay, so both the drugs were present, and it forms the micellar structure. Okay, we also determined the CMC of this dual drug conjugate, okay, by this pyrene assay method, and we found the CMC value 7.83. So if we disperse this conjugate above this concentration, so it will result into self-assembly. Okay, so the self-assembled nanoparticles we are prepared. So this is the AFM image, this is SAM, and these are the TEM images showing the self-assembled micellar nanoparticle, and the size was around less than 150 nanometer, around 124 nanometer was the particle size. Okay, we determined the in vitro release profile. Okay, so at different pH, like physiological pH as well as at the tumor interstitium pH, that is pH 5.4. So we found the release at pH tumor interstitium pH, okay, or tumor microenvironment pH, that is 5.4. It was relatively high as compared to the physiological pH, and the release were further augmented if we add the protease enzyme. Okay, so because in the tumor microenvironment these protease enzymes are present, so it digest the amide linkage. Okay, so it enhance the release of the individual drug. Okay, we also determine, okay, the stability, metabolic stability of the gemcitabine, which is very important. As I said, the gemcitabine rapidly undergo this metabolism into the inactive metabolite DFDU. Okay, so if we incubate the free gemcitabine, okay, in the plasma, okay, so within few hours, okay, this gemcitabine means if this study was conducted up to 24 hours, okay, so 62% of the DFDU was generated in the case of free gemcitabine. Okay, but in the case of this dual drug conjugate, okay, only 14% of the DFDU was generated. Okay, this is the in vitro stability profile we determined. So this we can conclude this metabolic stability of gemcitabine, it was significantly increased. Okay, we also checked the in vitro cell uptake. So we used two different cell lines. Okay, so one is MCF7, another one is the MDA MB231. Okay, so we took this coumarin 6 as a marker fluorescent dye. Okay, and the self-assembled nanoparticle, they are also loaded with this marker dye. So the uptake of the nanoparticle, micellar nanoparticle, it was significantly higher in both the cell line as compared to the free dye. Okay, and we also determined the in vitro cell cytotoxicity in both the cell line. So this self-assembled nanoparticle, micellar nanoparticle, they have a minimum IC50 value as compared to the free drug and their combination in the conventional form. Okay, in both the cell line. Okay, so it denotes that the cytotoxicity of this self-assembled nanoparticle, it was significantly increased. Okay, because might be because of the improved cellular uptake of the drugs. Okay, we also checked the apoptotic activity. So apoptosis is it is a desirable feature. It is programmed cell death. Okay, so we used the concept of differential staining. Okay, so this annexin V based kit we have taken. So what happened actually, the cell membrane, it contains phosphatidylserine from inside. Okay, so when the cell wall is damaged, okay, when the initial stage of the apoptosis, so when cell membrane start damaging, so that phosphatidylserine, it comes out onto the surface, or it is exposed onto the surface, which binds with the annexin V. Okay, so that gives the red fluorescence, and this green fluorescence, so these are the healthy cells. Okay, so the ratio of red fluorescence to green fluorescence, it is calculated by that confocal instrument software. So that gives the apoptotic index. Okay, so higher is the ratio, higher is the capacity of the apoptosis. Okay, so in the case of free drug, it is around like gemcitabine, it is 0.4. In the case of free docetaxel, it is around 0.5. In the case of free drug combination, the apoptotic index is around 0.75. But in the case of self-assembled this nanoparticle, apoptotic index was 0.98. Okay, the similar trend was observed in the other cell line. Okay, so we can say this self-assembled nanoparticle, it exhibit higher apoptotic potential. Okay, we also checked the in vivo pharmacokinetic. Okay, so we used this marketed formulation Gemzar and Taxotere. Okay, so we can see the rapid decline in the plasma concentration. But in the case of this self-assembled nanoparticle, okay, so for both gemcitabine as well as docetaxel, prolonged circulation can be seen. So it was the AUC value or the overall bioavailability PC. So it was around 5-fold increase. Okay, 5.3-fold in the case of docetaxel as compared to Taxotere, and 4.8-fold increase in the case for gemcitabine with reference to this Gemzar formulation. Okay, and we also checked its biodistribution by the same confocal microscopy. So diluted formulation we are administered to the mice, and we checked the we isolated different organs, and we imaged those organs in the under this confocal microscope. So as compared to this free dry and this self-assembled nanoparticle. Okay, so in the case of tumor, so very high drug concentration or high dye concentration can be seen. Okay, in vivo anti-tumor efficacy was also determined. Okay, so these images are self-explanatory. So in the case of nanoparticle treated group, we got the minimum or residual tumor burden approximately 30%, means almost 70% tumor was cured. In the case of this Fiji combination of this Taxotere and Gemzar, so the tumor burden was reduced only up to 40%, still 60% tumor was this. Okay, so of course I have not added that data. So we also did like morphological evaluation of the RBC. So these are the healthy RBCs, and these are the star shaped or pre-knitted RBCs we can see. Okay, these are also the shape is partially distorted. Okay, so the one is case with the Gemzar, that is the gemcitabine. Another one is because of this Taxotere, and this is with the self-assembled nanoparticle formulation. Okay, so this again looks like a healthy RBCs. Okay, means the hemolytic toxicity was significantly reduced. In the case of serum biochemical parameter like liver function test, okay, so the levels, okay, in the case of toxicity, the levels goes very high, okay, as compared to this normal or normal values. Okay, but in the case of our nanoparticle treated group, the levels we are almost comparable to the normal value. So we can say the toxicity level was very less. The similar trend was observed with nephrotoxicity parameters like blood urea, nitrogen, and serum creatinine level. The levels were very close to the normal levels. Okay, another case study I would like to present on this chemosensitizer and a docetaxel loaded albumin nanoparticle. So the problem with docetaxel, I already stated, it is water insoluble drug, and its marketed formulation contains dehydrated alcohol and a high surfactant concentration, P80, and it is also a substrate for this efflux transporter, P-glycoprotein efflux transporter. Okay, so its permeability is also less, means its cellular uptake, it is also less. Okay, so we thought if we can use some chemosensitizer like a quercetin. Quercetin is well-known antioxidant. Okay, so it will it also, quercetin is a well-known inhibitor of the PGP or any other efflux transporter. So it will increase the cellular uptake of the docetaxel. Okay, this was the first reason or rationale we took quercetin. Second reason, quercetin, it is well-known antioxidant. It also in high concentration, it acts as an anti-cancer or a cytotoxic. Okay, so or a pro-oxidant. So it can lead to synergistic anti-cancer efficacy, and it can also lead to reduction in the hepato or nephrotoxicity, which is because of the oxidative stress. Okay, so that's why we took this docetaxel and quercetin combination. And why this albumin nanoparticle? Because albumin it is also having multi-target mechanism. Okay, I will show in the next slide. Okay, this albumin nanoparticle multi-target mechanism. So albumin, it can leak into the tumor interstitium because of the enhanced permeability and retention effect. Okay, like if in the case of any inflammation or even normal injury, okay, albumin is the first thing which is that accumulate towards the inflammatory site. Okay, so that lead to swelling. So that is the very natural mechanism that albumin has tendency to accumulate at the leaky or this inflammatory sites. So cancer is not the exception. So albumin nanoparticle can easily go into the tumor interstitium via this EPR effect. And second thing, after reaching into that tumor interstitium, so this albumin nanoparticle interact with several receptors like GP60 receptor, GP18 receptor, GP30 receptor, spark protein, that is another receptor. So that is a secreted protein, acidic and rich in cysteine, spark. Okay, and VEGF receptor. Okay, so because of this multi-targeting mechanism, so it also internalized by the tumor cells. So it released the anti-cancer drug inside the tumor cell. So because of this multi-target mechanism, so we use this albumin nanoparticle. And the preparation of albumin nanoparticle, it is also very easy. So you might be aware that Abraxane formulation, okay, for paclitaxel, it is already available in the market. Okay, so that concept is Abraxane. Actually, Abraxane discovery, it was not a very planned discovery. It was a kind of like a scientist, we are studying the protein binding effect of paclitaxel, and they found when this paclitaxel was incubated with albumin, so that lead to formation of the nano aggregates. Okay, so that's how this Abraxane formulation was developed, and they modified the technology that is called NAP technology. So nanoparticle albumin bound. Okay, it is a patented technology. Okay, so using the same or almost with a little bit modification. So docetaxel, it also belongs to the same category like taxane category. So it can also form the nanoparticle with albumin. Okay, very easily because of the protein binding effect. So we used the same concept. Okay, of course we significantly modified the process because we also had to encapsulate quercetin. Okay, so but it was a very easy process to form the nanoparticle, and it is a scalable technology. Okay, so we got encapsulation efficiency of docetaxel approximately 75%, for quercetin around 68%. Okay, so and the size of the nanoparticle was around 200 nanometer. Okay, we checked the cell uptake. Okay, so again we used this fluorescent dye, that is a marker dye. Okay, so for free coumarin 6, okay, negligible uptake was seen, but this coumarin 6, which was encapsulated in the nanoparticles, significantly higher uptake was seen. Quantitative uptake was also determined. Okay, this is just the qualitative information. For quantification, we did not use this fluorescent dye. Okay, it was determined by HPLC analysis. Okay, for the this drug docetaxel. Okay, so approximately 2.5 fold increase in the cellular uptake was seen. Okay, as compared to free docetaxel. Okay, we determined the cell cytotoxicity analysis, like IC50 value was determined. So for in the case of free docetaxel, the IC50 value was 5.68, and if we compare at 72 hours, around 2.96. And in the case of self-assembly, this quercetin containing albumin nanoparticle, so it was after 72 hours, it was 0.71. So approximately 4-fold reduction in the IC50 value was seen. Okay, we also checked whether this docetaxel, it is also really a substrate for PGP efflux transporter or not. Okay, so we used this free rhodamine rhodamine as a dye, fluorescent dye. Okay, so it is a known substrate for PGP. Okay, so free dye, it was we did not see any uptake inside the cell, and free dye was incubated in presence of this quercetin, free quercetin. So little bit increase in the uptake can be seen, but it was not up to the mark. Okay, and rhodamine alone, it was encapsulated in the nanoparticle. Okay, so without quercetin, so again the uptake was significantly improved. But if we co-encapsulate quercetin also, okay, so the uptake was very high. Okay, we also checked its capability to induce apoptosis. So like in the previous case, so we got the highest apoptotic index in the case of quercetin or docetaxel co-encapsulated nanoparticle as compared to the free drug or the individual nanoparticle. Okay, for we also checked the PK profile. So PK profile like AOC value was also significantly increased. Okay, so around a 2.5 fold and 2.9 fold enhancement. Okay, as compared to this marketed formulation, that is Dosipar we have used. Okay, in the earlier case we used a Taxotere, but depending on the availability, so for this study we used this Dosipar as a marketed formulation. And anti-cancer activity was also checked. Okay, so in the case of like control animal, we can see okay significantly increase in the tumor burden, and at during means out of the 5 animals, 2 animals died. Okay, in the case of marketed formulation also 1 animal died during the study period, and the tumor burden was minimum in case of this docetaxel and quercetin encapsulated nanoparticle, and then no death of the animal was there. Okay, so percentage reduction in the tumor burden was approximately 65% in the case of our nanoparticle, and which was around 20% only in the case of marketed formulation Dosipar. Okay, we also checked the toxicity. Okay, so like this liver toxicity and nephrotoxicity, histopathology of the major organs, organ toxicity, hemolytic toxicity. So we concluded okay the formulation was relatively less toxic as compared to the this marketed formulation or free drug combination. Okay, now moving to next topic, that is nanomedicine for the oral cancer chemotherapy. Okay, so everyone will prefer that oral route is most easiest and convenient way of drug administration as compared to like parental administration. Okay, so but most of the anti-cancer drugs, okay, it requires they are not absorbed following oral administration. Not only anti-cancer drug, but many drugs. Okay, BCS class 2, 3, and 4 drugs, their bioavailability is a challenging task. Okay, so why the bioavailability, or what are the challenges the bioavailability or oral absorption of the drug, and how this nanoparticle they improve the bioavailability. So let's just spend 2 minutes time to explain the mechanism. Okay, so there are 2 different type of cells. They are present in our small intestine. One is called enterocytes and another is called M cells. Okay, so the normal drug molecules, they are absorbed by a passive diffusion from this enterocytes. Okay, so for this the drug need to present in the solution form, so dissolution of the drug. Okay, so if the drug is not water soluble, then its dissolution is poor. Okay, so its bioavailability will be less. Okay, second barrier is the permeability of the drug. Okay, so if the drug is extremely hydrophilic but it is not permeable, means its lipophilicity is very less, okay, then it would not cross this lipoidal membrane or intestinal membrane. Okay, so the permeability of the drug, it is also important. Okay, another barrier, if the drug is unstable in the GIT condition, okay, like highly acidic pH of the stomach and the presence of various digestive enzymes like proteolactic enzymes. So if the drug undergoes degradation under this acidic pH or because of this digestive enzyme, then again it would not be absorbed through oral route. And P glycoprotein PGP flux system, it also expressed onto the intestinal cell or onto the enterocytes. Okay, so the role of PGP is to protect. Okay, it is a protective effect, so it does not allow the absorption of the harmful or a toxin material. Like many times, okay, we eat street food which looks very tempting, okay, like golgappa or any other thing, chaat pakodi, okay, which looks very tempting, but it is not at all hygienic. Okay, but we do not get any infection. So we should thank to this PGP that it does not allow absorption of this harmful toxin or dust or this noxious material from the food ingredients. Okay, but at the same time, this anti-cancer drug and many other drugs, okay, so this PGP recognize that they will just kill the cell, they are harmful to the body. PGP cannot understand that these drugs are purposefully administered. Okay, so it does not allow the drugs to cross the intestinal barrier. Okay, so this is another problem. If the drug is substrate for PGP efflux, so it will not absorb. Another or last barrier is if the drug can overcome all these barriers, but first pass metabolism. Okay, so before reaching into the bloodstream, so major fraction is metabolized in the liver. Okay, so what happened if we prepare the nanoparticle of the drug or nano carrier of the drug? So encapsulation of the drug inside the nano carrier, it will protect the drug, okay, from premature degradation. Okay, like it does not directly expose the drug to the like acidic pH or digestive enzymes, so drug remain protected. Okay, and these nanoparticles, they are directly absorbed into the by the M cells by endocytosis process. Okay, so there is a no need for solubility, no need for permeability, because these are the direct particle taken up by the cells by endocytosis process. Okay, PGP also does not express onto the M cell, and moreover, this like polymer, lipids, or stabilizer like surfactants which are used to prepare the nanoparticle, so they inhibit the PGP activity. Okay, and from this route, M cells, the nanoparticle, they are absorbed into the lymphatic system, and from lymph they enter into the systemic circulation. So it also bypass the first pass metabolism. Okay, like this. So when free drug is administered, so either it will not absorb, okay, but if it can overcome all the barriers, it can need to undergo first pass metabolism. Okay, but when these nanoparticles are administered, so they follow the alternative path by lymphatic system, so it bypass the first pass metabolism in the liver. Okay, so in this way, this nanoparticle or nano carrier system, they improve the oral bioavailability of the drug. Besides this, there are several other mechanisms, okay, which are important. Okay, so how this nanoparticle improve the oral bioavailability, like this drug nanocrystal, okay, or nano suspension of the drug, so increase the wettability, increase the dissolution profile, so they increase the bioavailability of BCS class 2 drugs. Cytosine nanoparticle, they are reported to modulate the permeability or like a bioenhancer, so they modulate the tight junction protein or increase the permeability. Okay, lipid nanoparticle, okay, so they follow this cavule mediated transport. Okay, and this lipidic system mostly, okay, they follow this chylomicron pathway, so ultimately they are absorbed into the lymphatic system. Okay, so this case study I would like to present, this is self-emulsifying drug delivery system, okay, of tamoxifen and quercetin. So tamoxifen, it is a well-known anti-cancer drug for breast cancer therapy. So after surgical removal of the breast, okay, so tamoxifen is prescribed, okay, for 3 to 5 years of maintenance therapy. Okay, so but tamoxifen, the problem with tamoxifen, it is tamoxifen alone, it is BCS class 4 drug. So poor solubility, poor permeability. So to increase the solubility, tamoxifen citrate, that is water soluble salt, it is prescribed, but it can again undergo precipitation inside the GIT because of the common ion effect. Okay, it is a substrate for P glycoprotein efflux system, so it lead to poor permeability, and it also undergo extensive first pass metabolism in the liver, okay, during the absorption process. So it does not only decrease the bioavailability, but at the same time, during chronic administration, it develops or it causes hepatotoxicity. Okay, so this tamoxifen, because it is a we need to take, or patient need to take a daily dose of tamoxifen for 3 to 5 years. So it is not possible that he can go to hospital to take receive the injection of tamoxifen on daily basis. So tamoxifen citrate tablets, they are available in the market with reduced bioavailability. Okay, and as I said, the problem with tamoxifen, it develops the hepatotoxicity. Okay, because of the first pass metabolism in the liver upon administration, so that lead to like a free radical and lipid peroxidation. So oxidative stress induced hepatotoxicity. Okay, so that's why we thought we should include quercetin in the formulation. So n number of formulation approaches can be used to increase the bioavailability of tamoxifen like drugs, but we preferred this self-emulsifying drug delivery system. Okay, because it's a very simple process. It is nothing but it is a like a mixture of oil, surfactant and co-surfactant in which the drug can be solubilized. Okay, and then this oily mixture can be filled in the soft gelatin capsule shell like vitamin E capsule everybody might have seen, and either or alternatively we can also absorb the liquid formulation onto some solid carrier, and we can fill in hard gelatin capsule shell, or we can also compress the tablet. Okay, and upon oral administration, okay, so this form nanoemulsion inside the body without the need of any external homogenization or agitation. Okay, so this process, it's a very simple. It demonstrates high level of industrial scalability because no sophisticated instrumentation is required, and so many commercial formulation based on this concept, they are already available in the market. Okay, so we prepared this tamoxifen and quercetin co-encapsulated self-emulsifying drug delivery system. Okay, so which oil, which surfactant, which co-surfactant, it required lot of optimization. So we did all this optimization. We also did check the compatibility of these two drugs. Okay, tamoxifen and quercetin. Okay, and finally we prepared one composition, and all the excipients, oil, surfactant and co-surfactant, okay, which we have used, they are under within this IIG limit for oral delivery. And so upon oral administration, okay, so this formulation from nanoemulsion in the like 0.1 normal SGL, so size was around 115 nanometer. In the phosphate buffer, okay, pH 6.4, so the size was around 170 nanometer. So in both the cases the size was less than 200 nanometer. And the purpose of including quercetin in our formulation was to reduce the hepatotoxicity of tamoxifen. Okay, but we found this addition of the quercetin, so it is also increasing the bioavailability. Okay, so without quercetin, only tamoxifen sets, they increase the bioavailability by 3.89 fold. Okay, and with quercetin, the bioavailability was further increased to 8-fold as compared to commercial tamoxifen citrate. Okay, so initially we were surprised what can be the possible reason, because we have added quercetin just to reduce the hepatotoxicity, but later on we realized this quercetin, it is a PGP inhibitor, so that can be the possible reason. It is also helping in increasing the bioavailability of tamoxifen. Okay, so we checked the anti-cancer efficacy of this formulation. Okay, so this tamoxifen quercetin SNETS formulation, so that decreased the tumor burden to a very significant extent. Almost 80% reduction in the tumor growth was observed. Only 20% tumor was remaining in case of this tamoxifen marketed formulation. So tamoxifen, it is not a cytotoxic drug. Okay, so the tumor growth means only almost just 20 to 30% reduction in the tumor growth was observed. And in the physical form, like co-administration of tamoxifen with free quercetin, it was not effective because quercetin also has the bioavailability issues. Okay, and we checked the mortality of the animals, like we stopped the treatment after 1 month. Okay, but we monitored the survival of the animals for additional 1 month, so for total 2 months. Okay, so not a single mortality was observed with our SNETS formulation treated group. Okay, however, death was observed in the other control groups. Okay, we checked the toxicity also, hepatotoxicity, by measuring this AST level, ALT level, T-bars, and this GSH level. So as I said earlier, in the case of toxicity, the levels goes up or down as compared to the normal value. Okay, but in the case of our formulation treated, so the levels were almost close to the normal value. So we can say the toxicity was very less, okay, or negligible for our formulation. So for this formulation, which is a non-irritant, so water loss is significantly higher. Okay, so in the case of our HPV based gel or any other topical formulation, okay, water loss was not significant. Okay, from the histopathology of the skin also, we can say the capsaicin, it cause irritation of the skin, but other formulation, whether it is liposome or HPV based formulation, so the histology of the skin resembles to the normal or healthy skin. So just yesterday we received the mail in, okay, and this just yesterday this patent is also granted. Okay, so I will stop here. These are the key research area of my lab: multifunctional nanocarriers for targeted drug delivery. Okay, gene delivery via non-viral vectors. Okay, non-invasive vaccine delivery, oral bioavailability enhancement, topical and transdermal delivery, co-delivery of anti-cancer drugs for synergistic therapy, and lipidic products for effective drug delivery. So some of the aspect I have touched during my presentation. And these are the two books which I have published on targeted drug delivery. Okay, so this is upon invitation of Control Release Society, published by Springer. Okay, so along with Professor Padma Devarajan of ICT Mumbai, and this is authored book published by Elsewhere on cancer nanotechnology. And thank you very much for your kind attention. And if time permits, so I would be happy to answer some of the queries. Thank you.
Sir, we have question in chat box.
Yes, let me check. So one question is, what's the significance of this fluorescence ratio? So can you please clarify on regarding which slide you questioned? Ankita's question. What's the significance of this fluorescence ratio? And another question is, albumin nanoparticle can work by specific targeting also. So yes, I already mentioned it has a multiple targeting mechanism, and all these are the specific targeting mechanisms. Okay, GP40 receptor, GP30 receptor, spark proteins. Okay, so all these 3-4 specific proteins. Okay, so this is specifically binds with the albumin nanoparticle. So this is a highly specific targeting mechanism. And the non-specific thing is they accumulate at the tumor and microenvironment because of the EPR effect. Okay.
Dr. Sanyog?
Yes.
You've done an excellent job. Your talk was fantastic and you have done a lot of work.
Yeah, thank you, sir.
So I take the privilege to actually conclude your talk.
Yeah,
so,
I was actually reading through some of the questions. So of course, time is not there. Then students can write individually, or you can send these questions. So
quite better, I believe. But
yes.
one question is very interesting.
Yes.
Which one? I just reading that. This question is by some Priyansi Jain, student of so and so. He says that how nanobodies interact with cancer cells. Is this therapy is painful? Inquisitive question. Means
Yeah, actually, these nanoparticles we need to inject intravenously. So it's like you are taking any injection. So it is if you feel pain while taking any injection or vaccine, then it would be painful. Otherwise, it's not painful. And after reaching intravenous or after reaching inside the blood, okay, so they will interact with the receptor, okay, by like endocytosis process. So I'm not sure whether you have studied or not. I can see if I am having some yeah, this slide is there. So yeah, so like on the target cell, some specific receptors are expressed, on which normal cells these receptors are not expressed. And on the your multifunctional system, okay, some ligand molecule you have also entered. So when you inject, okay, so they will selectively binds with the target cell. They will interact with the receptor. Of course, this animation, some problem is there. So this they will interact with the receptor and they will bind with the target cell. But on the normal cell, these receptors are not there, so they will not bind. Okay, and after binding with the receptor, so they will be internalized by the cells. That process is called receptor mediated endocytosis. Okay, so of course, I am not carrying that slide that explain the receptor mediated endocytosis. While I teach to the students, so I have a very animated movie clip, so that explain how this receptor mediated endocytosis takes place. But that happens, and it is not painful.
okay.
Yeah.
So I believe those who have question or queries that they can directly email Dr. Sanyog so that he can better explain.
Yeah, my email ID is also mentioned on
Yes.
this title slide. You can note down.
So in conclusion, I can say that you've done a lot of work, and Dr. Jain has done a prepared oral formulation for delivery with the tamoxifen along with quercetin. Basically, this particular the co-encapsulated molecule, I believe, was intended to be incorporated to achieve the hepatoprotective activity, but however, that led to the formulation with higher oral bioavailability. Basically, this particular molecule helped in enhancement of the bioavailability of the tamoxifen. And the third and last part was on transdermal delivery with the doxtec cell. Okay, so the group has developed some high permission vesicles for the transdermal delivery of chemotherapeutic agent, and basically these particular sort of formulations are intended for check the various studies to show the superiority of the formulation, and lastly they perform the in vivo studies or in vivo tumor regression studies also, which showed that the formulation developed by them has superior efficacy in comparison to the natural drugs or the formulations. And in fact, he is this product one company also, the product related to or the formulation related to delivery of the tamoxifen. So I congratulate Dr. Jain for your excellent work in the field of nanodrug delivery system, and believe that all the audiences must have been benefited from your excellent talk.
Yeah, thank you, sir.
Thank you, sir, for your valuable words. Now I request Principal Sir Dr. Dinesh Kumar Mishra to facilitate our eminent speaker and chairperson.
Thank you, Neha. And thank you, Sanyog sir. It's always pleasure listening to you as you're working in the area of nanomedicine. And I remember when I was working there in the Department of Pharmaceutical Sciences, from at that time, we are seeing that Dr. Sanyog Jain sir is continuously working in this area. So I must compliment and congratulate Sanyog Jain sir for this nice work. And please accept our certificate of felicitation. So please accept, and thank you very much, sir. And thank you, Dr. Manish sir, because he has also spared valuable time. And he is also a renowned person in the area of nanomedicine who is working at CDRI Lucknow. So I request Neha, please share the certificate of Manish sir. Manish sir, please accept this certificate of appreciation. On behalf of entire organizing team and Indore Institute of Pharmacy, I would like to thank today's speaker and chairperson for sharing their valuable thoughts and sparing their valuable time. Thank you very much, sir.
Yeah, thank you once again, everyone. Yeah.
Thank you,
And
Dinesh.
let me tell you, the audience, it's there are 3 generations sitting over here, because yesterday the talk was started with Professor N.K. Jain, Dr. S.K. Jain, and today Dr. Manish Chaurasia sir is there, Dr. Sayed Jain sir is there, Sasank Singh sir is there, and I was also there. I was fortunate enough to work under these seniors. And today's second session will be taken by Dr. Rakesh Tikade. He is also alumni of Sagar. So I won't take much time, and Neha, please introduce today's second session chairperson and speaker.
Yes, sir. Our second eminent speaker is Dr. Rakesh Tekade, Associate Professor, NIPER, Ahmedabad. He is going to deliver the topic Prospectus and Retrospectus in Nanomedicine with Chairperson Dr. Shashank Kumar Singh, Senior Principal Scientist in Pharmacology Division at CSIR, Indian Institute of Integrative Medicine, Jammu. Sir has 19 years of experience in cancer drug discovery. Sir has worked with Win Medicare Limited R&D USA and its organization Modi Mundali Pharma Research Development Center, Switzerland, in formulation and development in process validation, Cadila Pharmaceuticals Limited, Ahmedabad. Sir has more than 100 international publications in peer-reviewed journals and 6 US EU patents in his credits. Sir has been awarded as Indo-US Fellow in 2009-10 by DST Indo-US Science and Technology Forum, New Delhi. His area of interest are cancer drug discovery from natural product, drug combination studies, targeted drug delivery system for cancer, and cancer immunotherapeutics. Now I request Dr. Shashank Kumar Singh sir to introduce our eminent speaker, Dr. Rakesh Kumar Tekade. Over to you, sir.
Thank you, Neha. Thanks for these words. At the outset of this program, indeed, it's my pleasure to be part of this symposia sponsored by AICTE and IPGA. And I would like to congratulate the organizer of IIP Indore for organizing on such a relevant topic so that it will be beneficial to the students. So at the outset of this program, I have been assigned a responsibility to introduce one of the next most eminent speaker, Dr. Rakesh Tikade. Dr. Tikade is presently he's working as an associate professor at NIPER, Ahmedabad. He has got more than 15 years of teaching and research experience. He has received his doctorate degree from Dr. Hari Singh Gaur, Central University, Sagar, in a split-side PhD Commonwealth Fellowship program from Leicester, Lancashire, UK. He did 2 postdoctoral studies. One is from University of Hawaii at USA, and another one was at University of Texas Southwestern Medical Center, USA. He is recipient of several awards and several fellowships like Commonwealth Fellow, AICTE, NDF Fellow, Irish PDF, CSIR, SRF, AICTE, JRF, and so on and so forth. He has worked as a Commonwealth Fellow at Centre Lancashire, Preston, UK. He has been got a industrial exposure also. He has worked with R&D Center at Ranbaxy Pharmaceuticals in India. He has recently been awarded as a Best Associate Professor Award. He has got a teaching experience of teaching PhDs, masters, bachelors at different university based in USA, UK, Malaysia, India. He is one of the most prominent scientists in the area of nanomedicine or nano research. He has been listed the top 2% scientist in the world, database released, published by the Elsevier in 2020-21. He has co-authored more than 300 publications to his credit. He has got 9 books edited with Elsevier and other peer-reviewed publication houses. He has got 4 patents to his credit. He has supervised more than he has supervised 3 postdoctoral fellows, 12 PhDs, and 70 master students. He has been to PI to several DST or ICMR sponsored projects. He is recipient of Pharma Innova Award by Troika Pharmaceuticals Limited. He has received various international grants from Fundicides Fund from Chile, National Science Center Poland, and from various science agencies from France and Iran. He is referee to many peer-reviewed journals, more than 30 international journals. His area of research is development of fit-to-purpose, translational, and novel target-specific nanobiomaterial. So with this small introduction, I would request Dr. Rakesh to present his work in front of audience. Dr. Rakesh, please.
Dr. Rakesh, please unmute.
All right. Thank you very much, Shashank sir. My pranam to you. Pranam, Dinesh sir. So
Thank you, Rakesh.
Thank you, sir. Thank you for this nice introduction, sir. May I please ask the coordinator to please allow me to share my screen, Dinesh sir? May I please share my presentation? Yeah. Currently, it has been disabled for some reason.
Please wait, sir, please.
Yeah. Okay, thank you very much. So, yeah, just a minute.
Your voice is a bit low. Can you just increase your volume?
Yes. Just a minute. Is it okay now, sir?
Yeah, now it is fine.
Am I audible clearly now, right, sir?
Yes, you are audible and we can see your slides also.
Okay.
You can just bring it to the presentation mode.
Yes, okay. Okay, just a minute. All right. So,
yeah.
So once again, thank you very much, Shashank sir, for this nice introduction, and thank you very much, Dinesh sir, for your invitation and this opportunity to again interact and crosstalk with the students, which I really ponder and admire. From my lecture discussion with the coordinator, Dr. Dinesh, I understand the background of the audience. So for them, today I want to focus my lecture specifically on ignition mode. So what is the core goal of my today's research is to ignite the youngsters, because today this platform is indeed monitoring the presence of future pharmacists of this nation, right? So being a future pharmacist, it's indeed your responsibility to take up the current challenges as well as the future opportunities, right? And one more thing, I am having sore throat, so please bear if I disturb myself with some coughs. So so again, to reiterate that today I want to ignite you future pharmacists. Okay, so to start with, can any one of you recognize these fibers? Okay, so these are the asbestos fibers, right? So these are the asbestos fibers. So from where you can see it at lots of places, right? The most common being as an insulators in cables, right? Those who have born before 80s, right, they must have seen the iron, right, which we used to iron our clothes, right? That time we used to change the coil of the iron by ourselves, and that time we used to happen to interact with this asbestos fibers, asbestos sheet, sub-zero during winter. So they place this asbestos in the wall. Okay, so they make a brick wall, they make following this, they add asbestos as an insulator, then they put wooden wall, and then they do plastering. Okay, so this is how they make their houses. Now come back to original preposition, why I brought this to my discussion today, right? So this is what's happening in Ukraine, right? Okay, just let me bring back my pointer. Okay, laser pointer is here. So this is what currently, right, it's happening in Ukraine, right? So there is a war going on between Russia and Ukraine. They are fighting with each other, and this is the current status of their houses, which are producing this debris. And this debris is nothing but a major chunk is being formed by asbestos. Okay, so asbestos is right there. From where these asbestos come? This asbestos come from the asbestos industries from the lower to middle income countries like India, Bangladesh, and Pakistan. Yes, we are the largest producer of this asbestos, India being the one. So this is the asbestos heap. You see how the soldiers in Ukraine are handling this debris, and you see how the workers in middle or lower income countries like India, this asbestos is being handled. Okay, so now what is happening because of this? Because of this asbestos is getting into the thoracic cavity of the laborers, right? ये हमारे लंग में चला जाता है. अभी लंग में जाने से what is the fate? It undergoes deposition. Okay, so definitely it leads to asbestosis or eventually carcinogenesis in, and this is very wide and common among the workers who work in asbestos industries, right? So question is, what are the factors that lead to asbestosis? So for example, students like you, if you happen to visit asbestos industry, or any visitor happen to visit asbestos industry, they do not get asbestosis. Why? Because there are certain reasons to it. What are those factors? Those, if you are visiting, you are getting a very minor doses, and our body has some protective mechanism. For example, if you inhale it, probability is very high that the mucus secretion of your lung cavity will expel it out. Duration, how long you are being there. Source, what is the source of your exposure? Transdermal, oral, nasal, what is the source? There are several other pre-existing factors. For example, is the individual is smoking or having some other liver disorder? On top of it, the major factor that governs the asbestosis probability is the size, shape, and chemical makeup of this asbestos fiber. So what is the conclusion of this discussion? Is that size matters. All matters has size. Everything that exists has the size, and if they exist, they has the some dimension, sizes. Okay, so in this context, okay, the material which are in nanoscale, they are being projected. Okay, for example, we are just discussing about the size of asbestos, and we are cross-talking with the disease asbestosis. So for example, if the size of, so this is very important, what is the size of the material, what is the size of dust particle, what is the size of pollutant. Okay, if you happen to visit Kishanpura pool of Indore, you will see that heap of dust being floating above its above that bridge. Okay, but what governs the fate, the biological fate, is the size. For example, if the size is approximately 10 micron, then the possibility is that the possibility is tremendously high that it will reach to the tracheobronchial cavity of the exposing individual. And that is the reason 10 micron size is considered to be the brim. Okay, above that, if the size is bigger than that, then our nasal cavity will expel it out. If you are considering the intra bronchial or tracheobronchial delivery or the lung delivery by inhalation, 2 micrometer aerodynamic diameter size of the particles are considered as the optimal. Okay, so the conclusion from this discussion is that size of any matter guides, okay, guides is biofate. Okay, now before this lecture, because I want to ignite you, I want to take you to the juncture of this research. First of all, I wanted to apprise you how this nano came into the picture. I tried myself to see or retrieve the documents related to the nano. Then I could find that the first application of this nano you can track back as a documented evidence in scriptures related to the Mesopotamian civilization. What is Mesopotamian civilization? I am so sure you are having a slant idea from your 8th or 9th class history that this is the civilization which has been flourishing around the river Euphrates and Tigris. This is that civilization which is currently based around Iraq, Iran, Turkey, in fact, Syria. So this is the place where first application of nanotechnology has taken place, and how this has been documented. They have retrieved one canister. This is that canister, and when they critically analyzed this canister, they found that the gold nanoparticle was plated on this. Still, it could retain its lustrous property, right? So this is the power of size of the particle. This is the power of size of the matter. So gold is same, but if you convert that into nanoparticle, its lustrous behavior still retains after several centuries. The same, when I was reading, I found that several emperors in India, they used to make first iron nanoparticles, metallic nanoparticles, and then they used to cast these nanoparticles in the form of their sword, and that is the reason they used to get a very powerful, very strong sword. That is the power of the particle size. In context to its application in modern age, it is the scientist called Richard Feynman, right? He was the first gentleman who first brought this terminology of nanotechnology through his book named as There's a Plenty of Room at the Bottom. I invite all of you to go through this book or go through the summary of this book. It's very interesting. Okay, so in his book, he narratively he mentioned that he just encouraged, okay, he encouraged the scientists in various fields, not only medicine but across whole field. He encouraged that if you can manipulate the materials at the atomic and subatomic level, then you can possibly unveil the powerful and intended property of the material, okay, than that of its conventional version. That was his suggestion from that book. Okay, and this is the book that is regarded as the foundation for the birth of the idea of nanotechnology. So what is nanotechnology, right? You have been hearing about this nanomedicine and nanotechnology since yesterday's. I am so sure you are aware, but just a refresher. This is whenever you are manipulating the structure in a novel way in its nano range, ranging from 1 to 1000 nanometer. And because of that change in its architectural composition, that you are having novel properties compared to that of the conventional shape or size or physicochemical properties. So conventional metal is there and a nanomaterial is there of the same. They are behaving differently. This is what exactly nanotechnology. Okay, so if you are building anything on an principle of manipulating the structure at nano range, it's called as nanotechnology. Now, if we ask where is nanotechnology lying today, so the simplest answer is it is lying everywhere in information technology, it is in energy sector, it is in food consumer sector. For example, if you track back your computer long back in Charles Babbage era when the computer was used to be the size of a room, Jacquard loom, right? Now, intermittently, the eventually the science progress, and then you happen to see the condensers, the integrated circuits to nano chips nowadays, right? Similar notions flown into solar sector, fuel sector, consumer sector. In fact, today you are seeing the concept of stain-proof clothes, waterproof clothes, or wrinkle-less or wrinkle-free textile. This is because of the successful successful conversion of conventional material into innovative nanomaterial. This is its power. And medicine is no more an exception, where we have you know uh, transfers tremendously. Okay, so this is what nanomedicine. So whenever you are applying the concept of nanomedicine to nanotechnology, okay, for repair, construction of any biological system, or to diagnose too, okay, and if these devices are serving on the principles of nanotechnology standard, this is referred as nanomedicine. So if you are building a medicine or diagnostic agent, okay, upon the principle of nanotechnology, simplistically, you can regard it as nanomedicine. Okay. Now, my simplest, I also want to, resolve the queries which I happen to hear from the audiences across different conferences, seminar. Okay. So are we seeing the nanomedicine in clinical trial, or it is just a lab-based research? So the simplest answer is yes, friends. They are, gigantically in the, they're gigantically in the clinical trial phase. Yes, there are several molecules, several products from several companies like Talon, Anzon, Xenius, Nippon, Samyang, Abraxis. They are doing many clinical trials. Okay. Now, next question, very predominantly asked: is there any nanomedicine, okay, available in the market? As also conversed by a previous speaker. So the answer again is yes. They have crossed the boundaries of lab. They have crossed the boundaries of clinical trial, and they have made their successful presence in the clinic. So they have been successfully transformed from bench to bedside. For example, Abraxane, which has received the FDA approval for the treatment of non-small cell lung cancer, for the treatment of breast cancer or pancreatic cancer, as expressed by previous speaker. Yes, they are protein or albumin bound nanoparticles. Similarly, Doxil is another nanomedicine which is already there in the market. It's a liposomal formulation. Okay. Or Onivide is another. Rapamune is another there, which is the nanocrystal. Okay. Cabenuva is another one which is used for the treatment of HIV infection. Okay. It is a nanosuspension based extended release formulation of cabotegravir or lalpivirine. Now, coming back to one important topic. Okay. So, are there any growing interest of the market? Is the market showing the interest toward the nanomedicine? My question. And the answer is lying with this slide. You can see this data that suggests that there is an growing dollar-based, right? It has reached several $100 billion investment by various companies. So the answer is yes. We are witnessing an era where the market is seeing an upsurge of nanomedicine investment. Okay. So are the companies coming forward? So the answer is yes. From the last 5 to 7 years, there has been tremendously grown interest of various pharma and non-pharma companies also. They are buying the shares of nanomedicine companies. Okay. Why? The simplest answer could be they are seeing the ray of hope. Future expectations are there. Future possibilities are there. And this is very important slide. You all are youngsters, future of, largest democracy on this earth. right? So this is an survey by Mordor, right? This is a, as per that survey, they say that currently we are around 9 to 10th economy of the world, right? We are approximately 2 trillion economy. India is 2 trillion economy. And Mordor says that by 2050, India is going to be the third largest economy of approximately 25 trillion. Okay. So we are going to be the 25 trillion economy by 2050. Now, this is the point. Now, this is the right time to question, are we really focusing on the right path? Okay, so now please have a look at this slide. This is a country-wise investment I pulled out. So if you see the country-wise investment of nanomedicine, right? So USA is the topmost country. They have invested like massive, followed by the China, Canada, France, Spain, Germany, and only the whole remaining countries except this six, whole globe is falling under this others bar, including India. So now, being a future pharmacist, you should ask yourself a question. Being this a platform for budding scientists, young researchers, I am so sure this platform is monitoring the budding and young scientists of this nation. Then we should crosstalk this to slide that we are aiming and we are aspiring to be the third largest economy by 2050. But where we are in the race of nanomedicine, we are nowhere, right? In spite of current efforts being made throughout our country, we are nowhere, because the data clearly delineates that on paper, on market, on investment ground, we are nowhere. This ignites a question: can we participate? Can we undertake the challenge of developing nanomedicine? Because yes, this is the future, because this is the current trend. You can see, okay, year-wise investment in $1 billion. Most of the companies, they are investing in cancer. Okay, so cancer is the area where nanomedicine is finding tremendous application and market potential, following this neuro, following that antimicrobial, following that anti-inflammatory, and then cardiovascular, and followed by this others. Okay, so this is the current trends in nanomedicine. Cancer nanomedicine is the prime focused areas of various investors as well as various companies. Okay, now from our India, right, it's not that efforts are not being made. No, we should be very optimistic, right? We should see how we can participate, and this is what our ignition question. Okay, so we are optimistic. Efforts are being made by the government of India also. For example, if you see Nano Mission, DBT, ICMR, they are every year calling for dedicated calls for proposal on nanomedicine. Okay, and I try to pull out ki kahan dete hain zyada funding. For your information, most of the funding goes toward drug delivery, right? To the drug manufacturing, API, we are relying more than 70% on China, which is not good. So to reduce that, there was a time in 70s when India used to be the exporter of API. Eventually, the trend got reversed. How? We do not want to get into that get into that question, but how we can participate. Okay, now nanomedicine application in diagnostic, in toxicological, in biomaterial, in imaging and medical devices are some of the prime sectors where these funding agencies support their fund. Okay, now let us try to understand ki yaar, with this nanomedicine, what new we are doing. We know that size matters. Size determines the biological fit. But before plunging into that research, we should understand the need of that research. Is that research is needed or not? Sometimes, there are several instances where people are solving a problem. When people are solving a problem, that technically doesn't need a solution. मतलब आप एक ऐसी प्रॉब्लम को कई बार सॉल्व कर देते हैं जिसकी की कोई सॉल्यूशन की जरूरत नहीं होती. तो let us first understand whether there is a need or not. So let us take an example of a parenteral administration, right? I want to understand कि भाई site of action पे कितना ड्रग जाता है. How much of the drug is going to the site of action? This is very important thing to understand, right? So let us take an example of, it is general, okay? So whenever you administer something, this is an example of drug onconase, which is used for the treatment of mesothelioma. Okay, 10% of the drug instantaneously goes to the plasma protein binding. 50% generally, 50% goes to various tissues, liver, lung, kidney, okay, other tissue. 25% gets fat bound. Plasma protein, circulatory protein takes 13% of it, and remaining 2% goes to the site of action. That is 0.2 mg out of 10 mg. So what is this signifying? That in general, only 2% of the drug reaches to the site of action in its conventional mode. The rest 98% is doing what? Is doing nothing but offering side effect, adverse effect, toxicity, and other complications to the body. And of course, we should not forget that majority of the cost of a pill is coming from the API. 98% of the API is not needed there, it's just 2%. But we have to administer with this high dose to build that flux, to build that concentration in the body, that the target intended site get 2%. Okay. So what targeted nanomedicine does? So if by conventional route, this is how drug distribution takes place. It distributes throughout the body. But the drug is needed in this case only here. It is a tumorous mice and only tumor site is in need. Okay. But in case of targeted nanomedicine, we are aiming, we are aspiring to take the material straight and preferentially to the site of action. This is what targeted nanomedicine. So what is this? This is superior advanced form of drug delivery, okay, where the pharmacologically active material is being selectively delivered to its site of action while sparing the normal cell, because these are not the normal cells, those need the treatment, right? So if you are, this is very important to understand, if you are the scientist who is aiming to develop the nanomedicine, the first thing to understand is to clearly delineate the ideal properties of the nanomedicine that you should deliver it to the patient. Okay, the first is that it should be inert, it should be non-immunogenic, it should be physically stable. ऐसा नहीं होना चाहिए कि if you are shaking it, it's no more nano range. It should be chemically stable. It should not interact with the excipient, it should not interact with each other, it should not interact with the biological fluids. Okay, it should restrict its distribution to the non-target cell, and that is why exactly you are developing it. So the distribution to the non-target tissues should be as minimum as possible. Okay, it should have a predictable and controllable release pattern. It should not affect the drug action. The way you are loading the drug in this nanomedicine, maybe you are physically loading, maybe you are conjugating it, maybe you are employing any other complexation technology to load your drug, but it should not affect the drug action. That means it should not affect the essence of that graph that we know from our first year B Pharm, with the concept of minimum effective concentration, the maximum safe concentration, and the therapeutic window. So it should not affect its drug action. Okay, and of course whatever drug loading methodology you adopt, okay, you can adopt a novel method to load the drug, perfectly fine, but it should not affect the drug efficacy. Probably your methodology is involving extreme pH utilization, it should not affect the stability of the drug. The drug should not get affected, maybe the temperature exposure whatsoever. It should not lead to any dose dumping. That means before reaching to the side action, it should not start releasing the drug, okay, or it should not dump whole drug at one go, because sometimes you are combining several doses in one dose, okay. So now if dose dumping happens, then probably the reversal becomes too tricky and difficult, okay. Of course, no drug leakage, it should not leak the drug. It should be biodegradable. It should not happen that the polymer you are using is getting accumulated at some portion of the body and then leading to some complications down the line, or requires separate surgery maybe, or separate detoxification procedure, which is not ideal, okay. And of course, the very first important, if you are trying to market your product, then your product should be scalable. It should be capable. For example, if you are making the dossier for regulatory filing, the first thing they are interested in knowing whether you can generate it, whether you can produce it, manufacture it on large scale as per the future market demand of this product, or simply you can just produce it on lab scale. It should be scalable, it should be reproducible, it should be cost effective. Then only you can meet the patient's expectation, okay? Not everyone are having paying capacity, so that ideally they should be cost effective, okay, right. Now this is one aspect which I was, previous speaker was also touching this. I would also like to, educate all of you on this. This is a very important phenomena. As per, one of the slides was saying that the current, sorry, the current focus of nanomedicine is in drug delivery. If you again bifurcate that drug delivery, you will find that this drug delivery in cancer research is the focused application area of nanomedicine. Why? Because if you connect nanomedicine with cancer, one added advantage of EPR, that's enhanced permeability and retention effect, come into the picture. So why that comes into the picture, you know very well, okay. You know very well, right? You are a student, right? Now you know that if the time is 5 minutes, 5 minute exam mein bacha hua hai, and you have to write a 4-mark question in 5 minutes, okay. So 4-mark question in 5 minutes, probably what strategy you will adopt is to write in rush, write in hurry, as against to your question number 1, right, which is of 2 marks and which you write in peace slowly. Now you compare the writing in first 15 minutes of your exam and last 15 minutes of your exam. You can clearly judge the delivery of quality of your handwriting. Definitely the question which you attempted in last 15 minutes may not be, most likely that it will not have the same appearance as that of the question number 1. The similar thing happens with the cancer also, right? Because in normal cell, the cell division take place slowly. So the cell and its offspring, newborn cell, they are the copy of each other, exact copy, okay. But in case of cancer, it is not the exact case, right? This is what happening here. I invite all of you to have an attention here. So these are the normal cell capillary, and these are the cancer cell capillary. So this cancer, they are producing this angiogenesis. This is the process called angiogenesis, when the tumor start growing, okay. Stage 1, stage 2, stage 3. This angiogenesis takes place when the tumor becomes 2 millimeters in size. That is the point where angiogenesis starts. So because of this angiogenesis, they start getting higher nutrition, higher fuel to multiply. Okay, so because of this, these capillaries are imperfect, having certain perforations. Okay, and these are sufficient enough to allow this nanoparticle to go through and reside into the, so enhanced permeability came from this. Because of this perforations, also called as fenestration, nanomedicine can go inside the tumor. And R stands for retention. Why retention? Because it has poor lymphatic drainage, so whatever goes inside stays there for a longer period of time. Okay, so this is all right. This is how the tumor looks like, right? This is the angiogenic cells. Okay, angiogenic blood vessels. Okay, so this is the normal cell, this is the angiogenic cells. Okay, so normal tissues, they are having tight junctions as against to that of the loose fenestration in cancer. So here the nanomedicine can release the drug selectively because there is a poor lymphatic drainage. Okay, now important thing for your information, God has created, checks, है ना, check लगाया हुआ है भगवान ने हर किसी को. Angiogenesis has to be there. If something cut, some damage happens in our skin, okay, angiogenesis has to take place, है ना, blood vessels regrowth तो होना ही है. But protein kinase G is an enzyme which is present in our body. It limit beta catenin. Beta catenin is the one which promotes angiogenesis, right? Now what happens in cancer cell? Cancer cell stop producing protein kinase G. Abhi protein kinase G is not there. No one is there to control beta catenin to undergo angiogenesis, and that is the prime reason. The first thing that cancer cell does is to inhibit protein kinase G. Protein kinase G expression is tremendously low, which is the anti-VEGF enzyme. Okay, so because of that, this happened. And this is one slide I would like to invite your attention. Okay, so this is normal injection of a drug, methotrexate, and this is nanoparticle directed injection of a drug. Now this is the scale. I hope you all know how to read it. So greater the, so if the uptake is higher, you will see more red, mid green, and low uptake is blue. So you can see here lower uptake of this happens when your conventional administration took place. But as against to this, if you convert that methotrexate into nanoparticle, you can see this is the tumor. Okay, this is the implanted tumor where you can see, blue. It has reached the injected ID per G of the tumor enhanced. You can see here the signal of drug significantly enhanced in the tumor when you apply it. Green bar is for the nanoparticle. So when you apply it as a nanomedicine. Okay, so this is about when you simply convert that methotrexate into nanoparticle. Matlab aapne Abraxane is there. Abraxane is albumin plus paclitaxel. Okay, NAP technology se you prepared a nanoparticle, but this nanoparticle uptake is passive. Passive kaise hoga? I just explained, EPR effect. But now there are ways where you can also make it actively target. How? By using ligands. Ligands jaise ki folate, transferrin, integrin, there are several. In fact, aptamers are also there. Okay, so if you conjugate or surface modify this nanoparticle with this targeting ligand, which are having higher expression in the tumor, you can further enhance the uptake, organ uptake. And this is one such example where transferrin has been coupled to the nanoparticle. Okay, so this blue yellowish is the one where transferrin coupled tetradine and daunorubicin has been administered to the mice, and significantly higher drug as well as significantly improved therapeutic outcome has been observed. Okay, so this narrates in simplistic manner that if you increase the targeting level, if you can increase the targeting index, you can improve the therapeutic efficacy, and this is what we call the level of targeting. Okay, so there are three levels. Okay, so there are three levels. If you can deliver the drug to the organ, for example, if the patient is having, say, liver cancer, but now currently what's happening, you are giving an infusion of the drug, that infusion is going throughout the body, so I regard it as zero order. Okay, now first order or first level of targeting, that means where you are successful in taking the drug to the organ. यदि आप organ में drug पहुंचा पा रहे हैं, you are attaining first level. If you are delivering the drug to the tissues, so for example, liver is there. If you are taking the drug to the liver, it is first level targeting. But the whole liver is not cancerous. It is only a portion of liver that is cancerous. If you can take the drug to the liver tissues which are cancerous, it is second level targeting. But it is exactly and precisely the cells of that tissue, cells of the cancer that are in need of that drug. If you can take your drug right into the cytosolic compartment of the cancerous cell or any target cell, it is called third level targeting. Now I invite all of you to ignite your thought process, connect this with the conventional therapy, and try to cross-talk yourself where our existing healthcare is functioning. हमारा current healthcare किस जगह function कर रहा है अभी, whether it is first level, second level, third level, what is intended. Probably you will reach to a consensus that if a patient is there, patient goes to the doctor, doctor today giving a pill, tablet, capsule, or even parenteral, it is reaching to whole part of the body. The whole part of the body is not in need of the treatment. Some portion goes to the organ, desired organ, some portion goes to the disease site. How much is that? We are just crosstalk is just 2%. So the 98% of the drug is just wastage, and that concludes the importance of nanomedicine. And this is where exactly nanomedicine is functioning and focusing. Okay. Our previous speaker was trying to explain the receptor mediated endocytosis. I was I was there and listening to that conversation. Probably because of time, he could not explain that. So I was having that slide. I just pulled out very quickly. So this is how, receptor mediated endocytosis of a targeted nanomedicine takes place. So these are the receptors. Okay. These receptors, these are movable. Okay, they are undergoing lateral movements. Okay, whenever any nanomedicine comes in contact, for example, folate targeted or dextran targeted, they undergo localization in one part. Okay, that's called the clathrin coat. There where it forms a concentrated receptor ligand interacted complex. Okay, and to it, adapter protein is the one, right, that makes it sure that only dextran targeted nanomedicine or dextran targeted molecule comes. Okay, then after some time, clathrin goes and attaches to it. When the capacity of that vesicle reaches to the maximum, clathrin pulls it inside, makes it into the form of endosome. This endosome then undergo rupture. Sometimes it also get translated into lysosomal compartment, and this is one more area of research where future research is because not all the endocytosed vesicle, they undergo rupture. A major chunk of that also goes to the lysosome, that leads to the drug loss. So this is one more compartment where the drug loss takes place. So this is how simply drug endocytosis takes place. After that, The clathrin coat again moves toward the surface, adapter protein again moves toward the surface, and this is how our physiology or cellular biomechanics maximizes the utilization of materials. Okay, so this is all about receptor mediated endocytosis. This is one more area where I just want to tell how we are working in the area of diagnosis, diagnosis and gene therapy. This is where our focus area of research, and one I will give I'm planning to give one example of gene therapy, how we have applied nanomedicine concept in resolving the delivery issue of gene therapeutics, right? How gene therapy act is by replacing the mutated gene, okay, or by introducing the functional gene, okay. So it is a process of silencing the expression of a targeted mRNA or targeted protein. They are very potent. I first happened to took training on this nanomedicine as a part of my postdoc in US, and that was the first time I could really see how potent they are as against to the conventional drugs, for example paclitaxel or methotrexate or doxorubicin, where they are IC50 are in nanomolar or micromolar range. The IC50 of this gene therapeutics are in picomolar range. You can imagine how potent they are. Okay, and one more thing, they are one of the safest alternative for API. Currently we are relying on China approximately 68%. Just today morning I pulled this out. So friends, today our government is trying to find alternatives. Alternatives for what? Alternatives for fossil fuels. For example, petrol. Alternative for petrol is now electric cars coming into the picture. Similarly, as per the mandate of Department of Pharmaceuticals, where the Central Minister Shri Mansukh Bhai Mandaviya, he is working enormously to find the alternatives for API. Today, you know very well that we are importing most of our API. We are tremendously depending on other countries, especially China, right? What happens if they just stop giving us API? Okay, not only our country, because we are selling the finished product dosage form to other countries, they will also be in problem. So finding an alternative for this API is in interest of everyone, and this is where exactly we have tried to apply nanomedicine. So what is the problem with gene therapy is that the RNA interfering or small interfering RNAs, they are short-lived. Their half-life is very short, 1 to 2 hours. The circulatory RNAs degraded. Another problem, they are negative in charge. Our cell membranes are also negatively charged. They cannot interact. You know very well, charged species do not show cellular uptake. Should be neutral, but they are negatively charged. That is the reason they show less transfection efficiency, endosomal trapping, off-target effect. So as a pharmacist, as a formulation scientist, basically this is the biotechnology problem which we collected, assimilated, and we thought, if this is your problem, why not this can be resolved by nanomedicine? So we tried developing a transfecting reagent. I'm happy to share that we have already completed this job. This was allocated to us as a DST SERV grant, which we have completed. We have filed 2 patents out of it, successfully completed its examination also. We are awaiting its approval, so both should come very soon now. So this is a dendrimer templated technology. You might have heard about dendrimer, where people are using it for delivery, but dendrimers, as soon as it comes, toxicity comes into the picture. So this toxicity, if you try to connect with dendrimer, you will see this is coming from its concentration and the generation, right? Generation matlab jitna jyada generation, more the generation, more will be the amine group. So we just cut down both, cut down the concentration tremendously, cut down the generation tremendously, and cut down the concentration both. So here what we did, very simplistic approach. For the first time, we applied dendrimer as a template loaded the gene onto it. With the help of this template, on this template, we developed the nanoparticle of albumin. Okay. Now, we have these are the references. I invite all of you to please go through it. These are in Nature Scientific Report as well as Molecular Pharmaceutics, which is in detailed description of what technology we have developed, and we have published it because we have already filed for patent, so there was no point in keeping it secret, or avoiding ourselves from any public description. So already it has been published. Please go through it. So now what is all about? Now from here on, I invite all of you to connect how we try to utilize small stuffs in resolving the big problem associated with gene therapy, right? So the first problem was key, it is not showing endosomal escape. We identified that dendrimer are one of the most potent excipient. We didn't use dendrimer as an carrier here. We used it as an excipient in extremely lower concentration. So what we identified that if you use dendrimer as a template, the net positive charge of the nanoparticle goes up. Now, because of that net positive charge going up, albumin is a negatively charged. Definitely it cannot be used to deliver genes. In spite of this, you can see several literature on gene delivery using albumin nanoparticle, but albumin less than 10 to 20% efficiency. But with this technology, we could reach more than 90% complexation technology by dendrimer templating. We could find that the zeta potential tremendously go toward the positive size. They are a nanometric size. Now, this is where, so the big problem was escaping the nanomedicine from the endosomal compartment. Students, if any nanoparticle doesn't escape the endosome into the cytosolic compartment, then eventually it will end up entering into the lysosomal pathway where harsh environment will degrade it, especially the genes. So you can see here, if you expose these nanoparticles to 7.4 pH, early endosome pH, late endosome pH, then you can clearly see that this Z average size of the particles tremendously goes up. If this goes up, we have identified that if it goes up, it creates positive pressure on the walls of the endosome, ultimately leading to its rupture. So that was the biomechanics of its action, where we employed simpler concept of dendrimer templating to overcome the problem of endosomal escape. Okay. Then this is the second thing about the serum stability. Okay. Where we identified that they are, because they are albumin, innate protein of the body, they do not react with the blood protein. So that is one good thing, that we could resolve the issue of serum instability. Right. This is another so the dendrimer which we use as a template, we went ahead to evaluate whether the effect of endosome affects the potential of this carrier. Then we identified, yes, if you expose it to physiological environment, they retain their potential because they are prepared at pH 7.4, but that doesn't affect the surface potential charge. If you expose it to lower pH, early endosome or the late endosome, we could clearly see that the pH goes up towards the positivity. Why? Because then we moved ahead to see the mechanism. Then we identified that it is because of the dendrimer protonation. Because if we expose dendrimer to different pH, if you expose it to acidic side, it will protonate. The terminal amine group of dendrimer undergo protonation. The protonation of amine group NH2, it forms NH3 positive. So because of this positive charge, this positive charge, they can repel each other. And because of that, it shows pH responsiveness. Then another big problem with genes is that it undergo degradation in the serum because of the RNase enzyme. So we evaluated it. This is the one with our nanomedicine which we developed, and this is the one, this is the positive sequence and this is the negative sequence. In the absence of RNase enzyme, they are perfectly stable. But in the presence of RNase enzyme, if you treat the gene with RNase enzyme, less than 1 hour it takes to degrade them. Okay. In case of the nanomedicine, it could retain it for more than 24 hours. We assessed it for 24 hours. We found.
So if audience has any questions to Dr. Fish, I welcome them. You can ask because of shortage of timings. You can email him, or you can, if you have any specific query, we can take up 1 or 2 questions. We would be happy to answer 1 or 2 questions. So I would request the audience that if you have any specific questions related to his work, we will be happy to answer your queries.
Yes, please.
So some questions are available in the chat box.
Miss Neha, one question we can take, because due to the lack of time, but any one question that you feel that it suits to the theme, definitely Dr. Rakesh would give the answer.
Yeah, we can take 1 or 2 questions because of shortage of timing. So any student has any specific question related to his work, like he has talked about the siRNA, gene silencing, and the endothelial mechanism of drug transport. So all these mechanism related or any material science related, if you have any specific query, you can.
So there is a question by Akshay Bhavaskar. Is it possible to treat breast cancer by applying the drug topically?
Yes. Okay. So on top of it, I would also answer one more question by Piali Jain after this. So first, let us answer the question of Akshay, right? He's asking, very good question, that can we treat breast cancer by topical delivery, right, Ms. Neha?
So, yes.
Yes. So why not? Why not, right? See, if your topical delivery can take your drug, permeate it across the dermal barrier, take it to the cancer site, why it cannot? So yes, the simplest answer is yes, it should be able to treat the breast cancer by topically applying the drug, but you need to make sure that the drug is penetrating through the dermal barrier and reaching to the cell. Few challenges will be here, okay, because you are talking about the topical administration. So you need to consider several factors, several factors, okay. For example, the molecular weight of your drug, okay. So number one is molecular weight, and the limitation of molecular weight concerning the transdermal delivery, okay. So if it is higher, say docetaxel, paclitaxel, vincristine, vinblastine, whose molecular weight are 800, 900, 899, then it will pose challenge as against to other drugs, say FU or some small size drug, okay. They can be easily permeated. So number one is molecular weight has to be judiciously considered. Number two, what strategy you are applying. Topical administration, simple solution can also be applied topically, but you need to consider whether your strategy is successfully capable of taking your drug to the target site. So are you considering the permeation sufficiently? If yes, then the answer to your question is yes. What else is needed in any disease? It's only the drug at its therapeutic concentration at the target site. If you can do this by any route, you can attain the therapeutic benefit. As in your case in breast cancer, if you are applying it topically, but your drug, if it is successfully permeating to the site of action, that is the cancer, breast cancer, you can definitely expect the therapeutic benefit. Now the second question is from Miss Priyanshi Jain from first year IIP. She is asking, if a person is suffering from cancer and we are giving nanomedicine, okay, so person is suffering from cancer and you are giving nanomedicine therapy, then what about normal cell? It will affect by nanotherapy or remain same? It's a very good question out of this today's discussion. So Miss Priyanshi, see, nanomedicine is not a magic. I want to clarify to everyone. If you are saying targeted drug delivery, please get rid of this conception that targeted nanomedicine whole drug to the site of action. No, it predominantly carries a drug to the site of action. Please carefully note it. So with nanomedicine, even I am considering your question as targeted nanomedicine. So Miss Priyanshi, if you are delivering the same patient with conventional drug delivery, then there is no question what is the exposure fate of the normal cell. Normal cell will definitely get the drug in similar concentration that of the cancer. While in the case of nanomedicine or targeted nanomedicine, the targeting index, हम इसको बोलते हैं targeting index, okay, or targeting capacity. Okay, so the targeting index means how much amount of the drug at the site of action considering the peripheral compartment. So that will be tremendously higher. It's not complete. It's tremendously higher in case of nanomedicine. So in case of nanomedicine, that is the reason in previous lecture you saw कि toxicity tremendously comes down. खत्म नहीं हो जाएगी, tremendously comes down. Okay. So as your question asked, कि will the patient normal cell will get affected? होंगे, उसको आप prevent नहीं कर पाएंगे. हर एक nanomedicine will not go toward the targeted cancer cell. Major portion of it will go. You cannot totally bar them from reaching to the normal cell. No, आप किसी भी targeted nanomedicine के पेपर को पढ़ेंगे, then you will see the biodistribution पे आप जरूर जाइए. सब लोग को मेरा आह्वान है, सब लोगों को मेरा रिक्वेस्ट है. I am requesting everyone to please go and check the biodistribution pattern of targeted liposome, targeted nanoparticle, targeted nanoconjugate. You will always see some portion of the drug is there in the liver, lung, kidney. लेकिन जब आप conventional से compare करेंगे, the bar is very big in case of nanomedicine. So the ill effect of the drug will substantially lower. That is my answer. I hope this clarifies, Miss Priyanshi. Thank you very much.
Thank you, Dr. Rakesh. Yeah, you have beautifully covered the topic, and you have very nicely explained the basics of biomaterial or nanomaterial, their physiological properties, how physico-chemical properties change with the changing the size of a particle, and how their biodistribution pattern get changed, how their bioavailability get changed. So all these parameters are very critical for designing any pharmaceutical preparation, because the specificity of a formulation is very important, and most importantly, like reducing the toxicity, and these nanomaterial plays very important role in controlling the toxic effect of a drug. So by doing this, certainly you can improvise the overall therapeutic benefit of any composition. So with this, I think we would like to close this session, and I would request to take over further session forward. So thank you very much, Dr. Rakesh, once again, and thanks to all the organizers for giving us this opportunity to showcase.
Thank you, sir. I'll take 1 minute. As a token of appreciation and a token of love, I would like to share certificate for Dr. AICTE as well as Dr. Shashank. So, Ms. Neha, please share.
Yes, sir. It's available.
Once again, on behalf of entire organizing team and Indore Institute of Pharmacy, please accept the certificate, Dr. Rakesh Tikade. Thank you very much.
Pleasure, sir. Pleasure.
For your nice presentation and elaborative discussion about the nanomaterials and nanomedicines.
Pleasure, sir.
Thank you.
Thank you, Sasank, sir.
Thank you, Dr. Dinesh.
Thank you, sir.
Thank you.
Thank you, sir.
Thanks a lot.
Okay.
Okay.
Thank you, sir.
Thank you, ma'am.
Shall we close now?
Yes.
Yes, sir.
Sir, our next session is poster presentation now. I would like to hand over the session to Ms. Saloni Yadav, ma'am, for further proceedings. Participants, be ready with your poster presentation.
video on है? Video on करो. ठीक. Okay.
Dinesh sir, can we start?
Yes, madam.
Very good afternoon to all the distinguished dignitaries and participants. I, Saloni Yadav, as Assistant Professor at Indore Institute of Pharmacy, extend my heartiest welcome on technical session day 2 of AICTE sponsored 2 days e-conference on translational research for nanomedicine organized by IIT in association with Indian Pharmacy Graduate Association, IPGA. Now, I would like to welcome our judges, Dr. G.N. Dharvikar sir and Mr. Preetham Siraskar sir. Sir is working as a principal in Acropolis Institute of Pharmaceutical Education and Research, Indore, and holds 22 years of academic experience. Sir has been the recipient of AICTE Junior Research Fellowship. Sir has guided many PG and PhD students, and Sir has published many research papers in national and international journals of repute. He has 4 patents published in his credit. His area of research are development of DCS-based novel drug delivery system, including transdermal, fast-dissolving drug delivery, development of modern herbal formulation, biopharmaceutic, and pharmacokinetic principles. Sir is a life member of many associations like IPGA and IPA, APTI. Now, I would like to introduce our next judge, Mr. Preetham Siraskar, sir. Sir is a manager in production at Glenmark Pharmaceutical Limited. Sir is having more than 15 years of experience in pharmaceutical industry. He has been successfully facing US FDA, MHRA, WHO Geneva, MCC, and Health Canada audit. Sir has actively participated in freight demo trials of various packaging related equipment. Sir has been awarded as Oak Star at Bokar. Sir has trained new supervisor for routine operation and documentation carried out in pharmaceutical industries. Now, before commencing to the e-poster presentation, I would like to brief about the guidelines to the participants. Each participant will be given only 2 minutes for your poster presentation and 1 minute for Q&A session. All are requested to strictly follow the time limit. All are requested to be ready with your presentation, and individual participant will be promoted as a panelist. So then only you will be able to unmute and present your screen. So let us start. Our first presenter is Shohinur Rahman. Shohinur Rahman, please unmute yourself and present your screen. Shohinur. Okay, the next participant is Shipra Mahal. Next participant is Himanshu Sharma. Himanshu Sharma, please unmute yourself.
Yes, ma'am. Yes, ma'am.
The next 3 participants are Miss Sunil, Miss Tanzeeb, and Shadab. Please be ready. Himanshu,
Yes, ma'am.
please present your poster.
I'm visible.
Yes, it is visible. You can continue.
Good afternoon, all of you. Respected sir, ma'am. My name is Himanshu Sharma. I am pursuing MPharm Research Scholar, School of Pharmaceutical Science, IFTM University, Moradabad. My poster presentation topic is effect of anandamide, a putative endocannabinoids in experiment model of inflammatory and neuropathic pain. First of all, I am discussing about the introduction. Pain is the uncomfortable sensations in the body. It stems from activation of nervous system. It caused by extreme or damaging stimuli such as burning of finger, alcohol etiquette. Pain can be generally divided into nociceptive pain and caused by tissue damage, and other is neuropathic pain caused by the nerve damage. Neuropathic pain feel like a stabbing, soothing, burning, or pickling sensations. Endocannabinoids are a class of compound which are clinically used in pain and various type of disorder. Recently reported that cannabinoids agonists play important role in pain by the activation of CV1 receptor. CV1 receptor is expressed in the peripheral system and central nervous system. In the present study, we have used anandamide material and methods. First of discussion about the first method, Caragen-induced inflammation. Group first, Caragen 1%. Group second, Caragen plus low dose of anandamide 25 mg per kg. And third group, Caragen 50 mg per kg anandamide. Group fourth, Caragen 100 mg per kg anandamide is high doses. And fifth group is Caragen 100 mg per kg into methazine. And second group discussion about the induction of neuropathic pain, CCI, which means of the chronic constriction injury. First of all, first group is induction of neuropathic pain. Group second is CCI, chronic constriction injury, 20 mg per kg, anandamide low dose. And third group, CCI 50 mg per kg medium dose and high dose 100 mg per kg. Group fifth, CCI 30 mg per kg pregabalin. And discussing the evolutions. First of all, plethysmometer. Plethysmometer is an instrument designed to measure the small changes in volume, usually displacement of mercury. Model second, mechanical allodynia. Mechanical allodynia is a painful sensation caused by an inducious stimuli like light and touch. Thermal hyperalgesia. Thermal hyperalgesia is a condition of altered perception of temperature. Describe highlighted sensitivity to nauseous, heat, or cold. Atheismometer is a device for measure the tactile sensitivity of the skin. The measure of degree of tactile sensitivity is called atheismometer. And final, I am discussing about the result. The present study demonstrate the analgesic effect of anandamide in neuropathic and inflammation pain models. Anandamide reverts the hypergesic effect of pain induced by CCI and carrageenan. First of all, I am graph represent and the high dose of anandamide 100 mg per kg. And second model, I am treating the schedule of chronic constriction injury. First day I am anesthetized 60 mg per kg ketamine, and then 3 days, 7 days after check the behavior parameters with the help of mechanical allodynia and thermal hypergesia. And last of all, 21 days, this is sacrifice the animal and then the histopathology and discussion.
Himanshu, your time limit is over. Please conclude.
Okay. After all conclusion, anandamide attenuates the development of experiment model neuropathic and inflammatory pain. The beneficial effect of anandamide may be due CB1 receptor and potential of ATP sensitive and potassium channel. Thank you.
Thank you, Himanshu. Sir, any questions or shall I continue?
Himanshu, hi, it's Pritam Siraskar.
Yes, ma'am. Yes, sir.
Why, as you have mentioned in this category in 4, and you have taken this model. So, what is the concept to select this particular model to evaluate this study?
Sir, I am evaluate the behavior parameters, all over behavior parameters, nerve injury. By the second model, chronic constriction injury, model.
Why you have selected this model for your study purpose? The criteria for selecting this model?
Sir, this is a given the my guide Shabazz Khan sir.
Okay.
Yes, sir.
Okay, thank you.
Thank you.
Himanshu, from where you got this?
So you're not audible. So your voice is not clear. We can't hear you. Okay, I'll continue. Next participant is Mr. Sunil.
Hello, I'm audible, ma'am?
Yes, you're audible.
The screen is visible.
Yes, it is visible. You can continue.
Good afternoon. My topic is exploring anti-cancer potential of
Your voice is not clear.
alkaloid camptothecin.
Sunil, your voice is not clear. Speak loudly, Sunil. We can't hear you.
As we know, the cancer is a life-threatening disease, which majority of the people suffering from it needs rapid cure
Sunil?
from it. As we know, lung cancer is the leading cause of disease almost now. Hello?
Yes, Sunil, we can't hear you. Your voice is not clear.
Now, sir? Now?
Yes. Now it is clear. Speak loudly.
Hello? Now,
now it is clear. You can continue. Next participant is Tanzeeb Rani.
Yes, ma'am.
Please present your screen.
Hello?
Sunil, there is some technical issues from your side.
I'm audible now?
No, we can't hear you. Good morning, respected sir, madam. I, Tanzeeb Rani, research scholar, under guidance of Dr. Shahbaz Khan from School of Pharmaceutical Sciences, IFTM University, Muradabad. I am going to present poster presentation on the topic pharmacological evaluation of Anthocephalus cadamba bark extract for hypolipidemic and anthelmintic activities. In this research, two models were used, hypolipidemic and anthelmintic. The animals divided into 7 groups in each model. In induction of hyperlipidemia group, first treated as normal control, group second treated as hyperlipidemic control, group third received hyperlipidemic plus bark extract 50 mg in low dose, 100 mg per kg in high dose by orally. Group 7 received hypolipidemic and standard drug fenofibrate 65 mg per kg orally. And for the anthelmintic activity, the standard drug piperazine hydrate 10 mg per ml, and after that biochemical parameters assessed. Result: the present study demonstrated that effect of fenofibrate and bark extract in hyperlipidemic model was found to be more significant. On other hand, the piperazine hydrate and bark extract have also potent action against helminths. Conclusion, the result obtained that standard drug showed significant role in hyperlipidemic activity as well as anthelmintic activity. The plant extract also showed more significant activity at the dose of 100 mg per kg in hypolipidemic activity, and at 50 mg per kg extract was found to be more active against the helminths. Thank you, ma'am.
Thank you. Sir, any questions? The next participant is Shadab Khan.
Am I audible?
Yes, you're audible. Present your post.
Ma'am, is it visible?
Yes, it is visible. You can continue.
Okay, ma'am. Good afternoon to all respected dignitaries, teachers, and my dear friends. My name is Shadab Khan, and I'm pursuing M.Pharm from Pharmaceutics branch from Indore Institute of Pharmacy. And today I'm going to present a poster on the topic Nano Cosmetics, Future of Cosmetics and Cosmeceutical Preparations. सबसे पहले हम ये जान लेते हैं कि कॉस्मेटिक्स और कॉस्मेस्यूटिकल प्रिपरेशन में फ़र्क क्या होता है, डिफरेंस क्या होता है। कॉस्मेटिक्स वो प्रोडक्ट्स होते हैं, वो आर्टिकल्स होते हैं जो हम टॉपिकली स्पेसिफिकली स्किन के लिए यूज़ करते हैं टू बी स्प्रिंकल्ड, टू बी पोर्ड और टू बी स्प्रेड ऑन द स्किन to enhance the beauty of our skin or to enhance the attractiveness of our skin. And a cosmeceutical preparation is that preparation in which all the properties of cosmetics are there, but an API, an active pharmaceutical ingredient, is also there to treat a specific degenerative disorder or skin disease. तो अब बात यह आती है कि हमारा जो थीम है कॉन्फ्रेंस का नैनो मेडिसिन का, तो नैनोटेक का क्या यूज है स्पेसिफिकली कॉस्मेटिक्स प्रिपरेशन में। तो नैनो टेक्नोलॉजी इज एन इनोवेटिव एरिया ऑफ साइंस दैट इंक्लूड्स द डिजाइन, कैरेक्टराइजेशन, प्रोडक्शन एंड एप्लीकेशन ऑफ मटेरियल्स, डिवाइसेस एंड सिस्टम्स बाय कंट्रोलिंग शेप एंड साइज एट द नैनोमीटर स्केल 1 to 100 नैनोमीटर। नैनोटेक एक ऐसी फील्ड है जो बहुत ही माइन्यूट लेवल पर जाके 1 से 100 नैनोमीटर की स्केल जो उसकी होती है वहां पर जाकर डिजीज को ट्रीट करने के में यूज होती है। To date, nanotechnology has been demonstrated to improve the performance of cosmetics in a number of different ways, such as several layers होती हैं हमारी स्किन की, जैसे स्ट्रेटम कोनियम, स्ट्रेटम ग्रैनुलोसम, स्ट्रेटम लूसिडम। तो कई बार टॉपिकल प्रिपरेशंस में उनमें को वहां से सेलुलर लेवल तक जाने में बहुत दिक्कतें फेस करनी पड़ती हैं। तो नैनोटेक इसमें भी हेल्प करता है। कंट्रोलिंग ड्रग रिलीज, ड्रग की रिलीज जो हमारे कन्वेंशनल डोजेज फॉर्म जो होती है उनमें जो हमें दिक्कतें फेस करनी पड़ती हैं ड्रग रिलीज को लेकर, वो इसको कंट्रोल करता है। इसकी ये एनहांस भी करता है फिजिकल स्टेबिलिटी को और मॉइस्चराइजिंग पावर को भी इंक्रीज करता है। और जो हम जब बाहर सनलाइट में जाते हैं तो यूवी की जो रेज जो हमें डैमेज करती है हमारी स्किन को, उससे भी हमें प्रोटेक्ट करता है। सो नैनो टेक्नोलॉजी कैन डेफिनेटली बी अ ग्रेट आइडिया टू बी अप्लाइड इन द फील्ड ऑफ कॉस्मेटिक्स। पर जैसे कि हमने अभी इस सेशन से पहले जो राकेश सर ने हमें बताया था नैनो मेडिसिन के बारे में, कि नैनो मेडिसिन कोई मैजिक नहीं है कि वो बिल्कुल ट्रीट ही कर देगा हमारी डिजीज को। नैनो मेडिसिन, नैनो कॉस्मेटिक्स ये कन्वेंशनल से अलग इस तरह होता है कि ये दूसरे जो नॉर्मल सेल्स हैं उनको अलग कर देता है, जो जिनको हमें टारगेट करना है उन सेल से, और स्पेसिफिकली उन टारगेट सेल्स को ही।
Your time limit is over. Please conclude your poster.
Okay, ma'am. So basically, I want to say that nanotechnology is very beneficial in the field of cosmetics, and it is a great field. It is a great opportunity for our scientists to deal with this new field of technology. Thank you. Thank you all.
Thank you, Shada.
Thank you, ma'am.
Our next participant is Tanvi Sharma.
Ma'am, it is audible.
Yes, you're audible. You can present your screen.
Yes, ma'am. Ma'am, it is visible.
Yes, it is visible.
Good afternoon, sir and ma'am, and my co-participant. I am Tanvi Sharma, and today I am presenting a poster. My topic is quality by design, modern approach to achieve a quality target. Quality by design is an approach that aims to ensure the quality of medicines by employing a statistical, analytical, and risk management methodology in the design and development of manufacturing of medicine. The aim of pharmaceutical companies based on the manufacturing innovation is a quality. The pharmaceutical company improvement in the efficacy of the product. Quality intentionally could be an idea used by Dr. Joseph Juran and believed that quality throughout the design into a product related to the means which a product was designed in the first place. Quality by design could be as a essence that eliminate the batch failure, minimize and expensive investing. QBD additional avoid the regulatory compliances issue and the structure level elements of QBD. Shaping the target product profile TPP is because of correlative of the quality, safety, effectiveness is the done identification of the standard characterization. The more medicines is a basic of the planning and development of pharmaceutical companies. Quality by design is a sweet science that offers a higher development and selection and
Tanvi, your time limit is over. Try to conclude your poster.
QBD methodology helps the characteristic and justify a TPP
Tanvi,
target product profile product. Hello.
Yes, Tanvi. Tanvi, your time limit is over. Try to conclude.
Yes, ma'am. Yes. Is QBD has a generate importance within the space pharmaceutical process such as drug development, formulation, analytical techniques, biopharmaceutical. The most reason behind the QBD is that the regulatory necessities as per QBD turn into a promising scientific tools. Quality assurance of pharmaceutical industry needs a regulatory compliance.
Thank you, Tanvi. Our next participant is Shohenur Rahman. Please unmute yourself.
Am I audible, ma'am?
Yes, you're audible.
Okay, ma'am. Hello everyone, good afternoon. I am Shohenur Rahman from MM College of Pharmacy, Maulana Ambala. My topic is control of various renal function by circadian rhythm.
Shohenur, we can't see your screen. Please present your screen.
Ma'am, is it visible right now?
No, it is not visible.
Ma'am, here is visible. It's maybe network problem. No, it's right now.
No, we can't see.
Let me 1 minute. Ma'am, right now.
No.
Is it okay, ma'am?
No, we can't see your screen. There is some technical issue from. Okay. Now it is visible. You can go ahead.
Okay, thank you, ma'am. Is it visible, ma'am?
Yes, it is visible.
Okay.
Go ahead.
Okay. My topic is control of various renal function by circadian rhythm revisited. Introduction. Endogenous rhythm that coordinate with the internal biological clock in a 24-hour time cycle powered by endogenous biological pacemakers are the subject of chronopharmacology. External synchronizer monitor and regulate circadian rhythms which are genetically determined, especially the daylight cycle. Chronopharmacology investigates how biological rhythms affect pharmacokinetics, pharmacodynamics, and toxicity, as well as whether administering a drug at different times of the day affects its pharmacological properties. Molecular mechanism of action. The determination of circadian rhythm is genetically determined by a core of set of clock genes, including 3 period genes. These genes and their products interload to from transcription translation reserve feedback loops that give the molecular basis of circadian rhythmicity during the day. Clock interacts with BML1 to activate transcription of the PAR and cry genes. Consequence in high levels of this transcript. PAR and cry proteins translocate to the nucleus and inhibit clock BML1 mediated transcription. During the night, the PAR cry receptors complex is degraded and the cycle starts again. Circadian rhythm in kidneys. Renal plasma flow, glomerular filtration rate, GFR, and tubular reabsorption and or secretion process have all been found to peak in the active phase and fail in the inactive phase in the kidneys. Notably, hypertension, chronic disease, kidney disease, renal fibrosis, and kidney stones are all linked to changes in the circadian cycle of renal function. Thus, renal circadian clocks may also
conclude
interact.
your session.
Yes, ma'am.
Time limit is over. Conclude your session.
Okay, conclusion. Disrupted circadian rhythm is in kidney as a result of increased age likely contribute to adverse health outcomes such as nocturia, hypertension, and increased risk of stroke, cardiovascular disease, and organ failure. Ma'am, that's it. Thank you, ma'am.
Thank you, Shoinur. Our next participant is Ms. Shivani Burman. Ms. Shivani. Next participant is Ranjit Kumar. Ranjit. Next participant is Nargish Jahan. Mr. Alok Kumar Mishra.
Hello.
Yes. Nargis.
Good afternoon, ma'am.
Yes. You can present.
Okay, ma'am.
Nargish, present your screen.
It's visible.
No, it's not visible. Yes, now it is visible.
Okay, ma'am, दिखा रही हूँ बस. Visible, ma'am.
Yes, you can continue.
Okay, ma'am. Good afternoon, respected sir and ma'am. I am Nargis Jahan. I am pursuing M Pharma. Currently, I am M Pharma from CET College of Pharmacy. My poster is representing polyherbal formulation as a medicine. And abstract medicinal medicinal and therapeutic effect of herbal formulation, that is a polyherbal formulation, has been widely applied in the treatment of various disease in all over world due to its benefit along with easily availability and in naturally herb combination. And introduction dependence of human beings on plants dates back to the origin of the human race. The basic needs of life for human being are shelter, clothing, food, flavor, and fragrance, and not the least medicines. Plants are a common source of medicines. Plants have saved the creation of refined traditional medicines structures, among which are Ayurvedic, Unani, and Chinese are common. And Indian, Chinese, Roman, and the Greek civilization plants were the very foundation of the care systems and deemed to possess a divine and a supernatural power of healing. That is a traditional medicine, herb combination, drug combination, polyherbal formulation. And effect of herbal formulation, that is a polyherbal formulation. So, and conclusion में मैम many herbal therapeutic are still under in vivo evaluation and have not been evaluated by clinical trials. There and then there is a needed of time to evaluate polyherbal formulation using a scientific methods such as a clinical trials, possible bioactive compounds, and a mechanism of action for the future world. Thank you, ma'am.
Sir, you're not audible.
Nargis, I have one question regarding this polyherbal formulation. Have you, in your literature survey, have you find any other reference document where it was showing that polyherbal medicine having a greater effect than other formulation available in the market?
Can you speak, sir?
My question is, as per your poster presentation, have you during your preparation, have you find any research paper who is exploring that your polyherbal formulation is better than other formulation available in market? Okay, no problem, no problem.
No, sir.
Thank you.
Okay, sir. Thank you, sir.
The next participant is Mr. Alok Mishra. Next participant is Mohammad Shoaib. Mohammed Shoaib, unmute yourself.
Yes, ma'am.
Please present your screen.
It's visible, ma'am.
Yes, it is visible. You can continue.
Good afternoon, everyone. Good afternoon, ma'am and sir. So I'm Mohammad Shway from Shiat College of Pharmacy, Varanasi, and student of M.Pharm Pharmacology. And my poster presentation topic is pharmacological accept of Quisqualis indica lean and its medicinal properties. So it's a my drug is Quisqualis indica. It's a herbal drug, and we use its leaf and for treatment of rheumatoid arthritis like diabetes, heart disease. When we treated herbal biotics considered as promising more suitable due to its minimum and no side effect. We use herbal drug because it's a low side effect and it's
Mohammed, you are not audible.
hello.
Yes,
Audible.
Mohammed. Yes. Conclude your poster. Your time limit is over. Try to conclude.
My drug is Quisqualis indica. उसमें हम ये बहुत सारी एंटी इन्फ्लामेशन और एंटी इम्यूनोमॉड्यूलेटरी एक्टिविटी शो करते हैं और मैम ये इजीली अवेलेबल होते हैं इंडिया के हर रीजन में। इसको मैम इसके ड्रग को मैंने यूज किया है रूमेटाइड आर्थराइटिस ट्रीट करने के लिए एज ए इम्यूनोमॉड्यूलेटर एक्टिविटी इन विजिटर रैट।
Mohammed, you are not audible. Have you concluded?
Yes, ma'am.
Okay, then stop sharing. Is there any question or shall I move forward? Next participant is Mr. Alok Kumar Mishra. Alok,
Yes, ma'am.
Yes, present your screen.
Just wait a minute. I'm presenting my screen. Is it visible to you, ma'am?
No, it is not visible.
Oh, no. Is it?
No.
I have shared my screen. Still, it's not showing, madam.
Alok, your screen is not visible.
Just just give me a minute, madam. Say. Now is it visible, madam?
It is not visible.
Set my screen. Why it's not visible?
Are you able to present or will move forward?
I don't know why my screen is not visible to you. I have said it already.
No, we can't see.
Can I present only, madam? Just
Okay, so our next participant is Mr. Rakesh Kumar. Rakesh Kumar. Next is Mr. Harshvardhan Bilore.
Yes, ma'am.
Unmute yourself. Yeah.
Ma'am, I'm audible?
Yes, you are audible. Present your screen.
Okay, ma'am. Good afternoon to respected sir and ma'am. I am Harshvardhan Bilore. My topic of presentation is...
Harshvardhan, please speak loudly.
Yes, ma'am. My topic of presentation is an overview on Emulgel, a cutting-edge drug delivery method for the delivery of hydroprophic drug. As you all know that topical drug administration is a localized drug delivery system anywhere in the body through ophthalmic, rectal, vaginal, and skin as a topical route. These are applied a wide spectrum of preparation for both cosmetic and dermatologically to their healthy or diseased skin. As we know, the advantages of gel, a major limitation is in the delivery of hydroprophic drug. So to overcome this limitation, an emulsion-based approach is being used so that even hydroprophic drug moiety can enjoy the unki property of gel. When gel and emulsion are used in combination of doses form are referred as emulsion. In recent years, there are being great interest in the use of novel polymers and uncu accept of dermatological pharmacology in the direct accessibility of skin as a target organ for diagnosis of treatment. Within the major group limitations, group of semi-solid preparation, the use of transparent gel has expanded both in cosmetic and pharmaceutical preparations. There are certain advantages of emulsion, such as incorporation of hydroprophic drug. The hydroprophic moiety cannot be added directly to the gel base because of the improper release shown by the drug as a lack of solubility. The emulsion allows the addition of such hydroprophic drug in the oil phase, which leads to the dispersion of oil globules in aqueous phase, resulting in the formation of oil in water emulsion. Further, this emulsion can be simply added to the gel base, thereby providing good stability and better release of drug. It has better loading capacity, better stability, production feasibility, and low preparation cost. There are certain disadvantages such as it causes skin irritation, some allergic reaction. It has poor permeability of drugs, some drugs to the skin. Now moving further to the materials and methods. Materials used such as aqueous materials, water and alcohol, oil such as light liquid paraffin, emulsifiers such as 2-in-80, sodium stearate, gelling agents such as HPMC, penetration enhancers, formulation of emulsion. Formulation of emulsion. Emulsion are prepared by incorporating gel and emulsion. The emulsion are prepared separately and mixed together. For preparing emulsion, aqueous phase and oil phase are taken separately and mixed. Then the gel is prepared by using gel agent. After preparing gel and emulsion, they are mixed with stearin. The aqueous phase is prepared by mixing 280 and water. The drug is dissolved in ethanol and the two phases are mixed by continuous stirring. After preparing an emulsion and gel, they are mixed together to get emulsion. That there are certain evaluation parameters such as physical examination, determination of pH, rheological study, spreadability, swelling index, in vitro yeast study, stability study. In result,
Harshvardhan, your time limit is over. Please conclude and speak loudly. You're not audible.
after a thorough literature survey, I conclude that emulsion has proven to be one of the most effective, better, and convenient drug delivery system, as has ability to deliver hydrophobic as well as hydrophobic drug. Thank you.
Harshvardhan, what is the stability for the product? Stability time, which is reported?
Sir, it's stability 6 hours, sir.
6 hours. No, that is your sustain release time period. I'm asking about if we will manufacture this kind of product. So, what will be the stability? Are you getting my point?
Yes, sir.
Okay, no problem.
Thank you, Harshvardhan. All participants are requested to please strictly follow the time limit. Our next participant is Mr. Yash Rauka. Unmute yourself.
Am I audible?
Yes, you are audible. You can continue.
Screen is visible.
Yes, it is visible.
Good morning. My name is Yash Rauka, pursuing BA from second year from Indore Institute of Pharmacy. Today, my poster topic is application of nanofabric. Aim of nanofabric is to form protective garments, especially for emergency service workers, such as military, firefighters, medicinal personnel, etc. Nanofabric can be used to prevent cancer caused by UV rays, respiratory chronic disease, as we see in COVID-19, and also from bacterial infections by wearing masks, gloves, suits, shoes, coats formed by nanofabrics. Nanoparticles used in fabrics are silver nanoparticles. Silver nanoparticles acts as an antibacterial, while zinc oxide, titanium dioxide acts as a sunscreen due to high absorption of UV rays and makes fabric UV blocker. These nanoparticles makes the fabric so dense that the bacteria, even UV rays, can hardly penetrate, as shown in the evolution. These are so many methods of nanofabrication shown in the chart, in which widely used in electrospin. At the last, I want to conclude that the nanofabric improves properties such as UV blocker, antibacterial, antiseptic, and enhance the performance. It is having lightweight with a small diameter that improves blood circulation and reduces consequently sweating. Thank you.
Thank you, Yash. Our next participant is Miss Ayushi Arora.
Am I audible, ma'am?
Yes, you are audible.
Is my screen visible?
Yes, you can continue.
Good afternoon to all the dignitaries present here. Myself, Ayushi Arora, an undergraduate student from Indore Institute of Pharmacy. I'm here to present my e-poster on the topic nanotechnology-based cosmeceuticals. The word cosmeceuticals are made up of two words, cosmetics and pharmaceuticals. So cosmeceuticals are cosmetic products incorporating biologically active ingredient giving therapeutic effects. And when various nanomaterials have been used for the development of cosmetic products, then they are known as nanocosmeceuticals. With due increase in the market for personal care, The cosmeceuticals are contemplated as the fastest growing part of the personal care industry, and an increase in the cosmeceutical are being crossed over by novel drug delivery systems. The advantages of nanocosmeceuticals are controlled release, site-specific, physical stability, active transport of API, increased efficiency. As you can see here in the pictorial representation, the different types of nanocosmeceuticals are nanoemulsion, nanocapsule, neosomes, solid lipid nanoparticle, liposomes, transferosomes, and dendrimer. These all types of nanocosmeceuticals have their own advantages for formulating the product, but at the end, they all provide the advantages as we discussed earlier. I want to add some examples that the brands that are using nanotechnology for making their products, like L'Oreal is making an anti-wrinkle cream using nanotechnology. And Antria, a brand, is making a sunscreen. Sinair is making an hair growth product. Marie Louis Cosmetics is making a nutrition cream. And Lifeline Skincare is making an anti-aging cream. So I would end my presentation by discussing the future perspectives of nanocosmeceuticals. With the increase in demand of cosmetics among the society, it is an industry which will see a boost in the market in the coming future. It is the only industry which was first to use the principles of nanotechnology in their product development. With the current trend in increase in the research and development related to the use of nanotechnology in cosmetics gives the indication of the advancements of its future success, development, and acceptance. Thank you.
Aisha, I have one question. Is this preparation by a nanotechnological based cosmeceutical will be the affordable for the patient or customer?
So right now the branding. Yes, so right now the products are at a higher cost because of the branding and everything. And nanotechnology is not very effective, especially in India. So, but in the coming future, we are expecting that as the nanotechnology grows further, the costing would be reduced.
I must say it's a good presentation. And Yash, also you have presented very good presentation and topic. Thank you.
Thank you, sir.
Thank you, Ayushi. Our next participant is Rahul Bharti.
Yes, ma'am.
Present your screen.
I am visible?
Yes, your screen is visible.
Okay. Good afternoon, everyone. My name is Rahul Bharti. I am M.Pharm first year student of Smriti College of Pharmaceutical Education, Indore. And my today's topic is aptamer as a future drug. And then what is aptamer? Aptamer are a nucleic acid molecule that mimic the antibodies by folding into complex 3D shapes that bind to the specific target. Some aptamer exist naturally as the ligand binding elements of ribo switch. Most aptamer generate in vitro and can be tailored for a specific target. And use of aptamer, application of aptamer technology in therapeutics, in new drugs, in diagnosis of the disease, and in new drug delivery system and bioimaging. Selection of aptamer. The selection of aptamer is done by cell select technique. Select means systemic evaluation of ligand by exponential enrichment. For target drug delivery, it is used as aptamer drug conjugate for gene therapy, aptamer drug conjugate for chemotherapies. There are several aptamer formulation under clinical trials in which mucogen, pegotinib are under the phase 3 clinical trials approved by the US FDA. Then conclusion of the it has been over 2 decades since aptamer are first described. Multiple unique feature of aptamer arouse extension interest in the development of the aptamer as a nucleic acid based alternatives to antibodies and peptide ligand for the versatile biomedical application ranging from in vitro diagnosis, molecular imaging, drug delivery to biomarkers, discovery and drug screening. Preclinical investigation of aptamer have shown promising efficacy as well as safety profile. Aptamer provide a promising and unique tool for therapeutics. Give the fast progress made in the field. It seems like that in the next decade, aptamer may be a common instrument for the clinical practices. Thank you.
Thank you, Rahul.
Yes, ma'am.
Our next participant is Ms. Sapna Saini. Ms. Sapna?
Ma'am, am I audible?
Yes, you are audible. Please present your screen.
Yes, ma'am. Ma'am, it is some technical issue. Ma'am, my screen is visible?
No, it's not. Sapna, try to present your screen.
Yes, ma'am, I'm trying.
Are you able?
No, ma'am. It's just
Okay.
some technical issue.
Okay, we'll take another participant. Ms. Ankita Bhadoria.
Am I audible, ma'am?
Yes, you're audible. Ankita, please present your screen.
Ma'am, is it visible screen?
Yes.
The poster is visible?
No, the poster is not visible.
Now?
Yes, it is visible. You can continue.
Good afternoon to all the dignitaries and my co-participants. I am Ankita Bhadoria, pursuing my M Pharmacy in Pharmaceutical Quality Assurance from Indore Institute of Pharmacy. I would like to thank our respected principal to giving us such a great opportunity for this interactive session. Thank you, sir. Today, I'm going to present my my topic of Six Sigma, a quality improvement methodology. As we are aware that the quality is vital for every industry, but it is necessary for pharmaceutical industry to maintain a quality product to give customer satisfaction. And there are so many tools which are contributing to maintain the quality, and industries are adopting it as well. In this tools, one of the tool is Six Sigma, which is also a quality improvement tool working to prevail excellence in the organization. It is a set of numerical and statistical tools which works mainly based on the principle of customer focused teamwork, to provide flexible approach, reduce the product reduction, and identify the root cause of the problem. This is mainly based on the
two methodologies of DMAC and DMA-DV. Being a statistical method, there are so many statistical quality control tools like control charts, histograms, scatter plots, and check sheets, which will help in determination of the root cause and also in improvement and taking the major control to improve the product quality and define the customer satisfaction. Now there are it sets a quality indicator as it is prominent to deliver the product which is less than 3.4 defects, producing less than 3.4 defects per million, and an individual which can contribute or which can sustain in this methodologies can be pursued some belts or certification in the form of white belt, yellow belt, green belt, black belt, and master black belt as well as being a quality improvement tool. It having so many advantages of cost and project time reduction, development in process design, product development and process design, and it improves and control the product defects. At last, being it as this tool product ensures the in delivery of near perfect product. So with the advancement of this tool in the future, it may be a very effective tool to control the quality of the product. Thank you so much.
Ankita, first of all, I would like to say your topic is very good. I'm not expecting this kind of topic from you people, but I'm quietly surprised that and want to say ki you are very in a good hand and very fantastic topic you have selected. Congratulations to you.
Thank you so much.
One question from my side.
Yes, sir.
Is this Six Sigma methodology can be implemented for a pharmaceutical only, or you can implement it anywhere?
No, sir. This methodology, as being a quality improvement methodology, we all know that quality is important for every industry, and the starting of this methodology is done by Motorola, which is a high-tech industry. And when the performance of the Motorola is declining in front of their Japanese competitors, they have only invented Mr. Smith, the scientist and engineer in the Motorola, have invented this improvement tool to reduce the product defect and deliver a quality, so that many IT industries, technological industries, and now healthcare industries in the current trend is adopting this technology, so it is applicable for others also.
Fantastic. Thank you so much.
Thank you.
Thank you, Ankita. Our next participant is Mr. Neil.
Hello, am I audible, ma'am?
Yes, you are audible.
Is my screen visible?
No, it's not.
Okay, wait. Is it now?
Yes, it is. You can continue.
Good afternoon. Good afternoon to respected sir and respected ma'am and all my fellow participants. I, Neil Ravi Daniel, currently pursuing my master's in the field of pharmaceutical quality assurance from Indore Institute of Pharmacy, Indore. And my today's topic for presentation is quality metrics as performance indicator. What is quality metrics? Quality metrics generally refers to a set of standards set by the regulatory authorities which are required for the application or for the delivering of a safety or of a standard product which will not affect the health of an end user, it means patient. Quality metrics are the measurements of the value and performance of the product and services and processes which are being provided by the industry, mainly for the pharmaceutical industries. Quality metrics are refined and systematic representations of quality key performance indicators during manufacturing operations. Because we cannot put quality directly into the end product, we have to maintain it throughout the production, throughout the starting raw material process and into the end product. Pharmaceutical measures include in key performance indicators reporting come from research, development and manufacturing and distribution and actions. Key performance indicators. Key performance indicators can be defined as those indicators which works in combination to deliver the quality product to the end user. They enable us to understand the performance and health of our business that can make critical adjustments in our execution to achieve our strategic goals. There are majorly 7 types of quality metrics, which can be defined as performance, features, reliability, conformance, durability, serviceability, and aesthetics of the industry. And by all of this data, the FDA is currently approaching for the risk-based regulatory approaches, in which they collect the data from the pharmaceutical industry of the batch recalls of defects, of deviations, and each and everything, through which they can statistically or they can graphically present the quality of the product they are meeting or not. By this, we can conclude that quality metrics play an important role in the desired state of pharmaceutical quality and regulation, and it supports the continual improvement of the pharmaceutical quality system. And it is not a unit operation of any single personnel. Each and every unit in the pharmaceutical industry has to work throughout and in combination to give the quality product to the end user. Thank you, ma'am. Thank you, sir.
Nice topic, Neil.
Thank you, sir.
Thank you, Neil. Our next participant is Ms. Janvi Yadav. Please unmute yourself. Janvi.
Hello. Am I audible, ma'am?
Yes, you're audible.
Yes, ma'am. Ma'am, there is some problem in screen sharing. Hello?
Yes.
Ma'am, actually there is a problem in screen sharing.
You are unable to share?
Yes, ma'am.
Okay, we'll take next participant. No, now it is visible. We can see your screen. Okay, we'll take next participant, Miss Advita.
Yes, ma'am.
Yes, please share your screen.
Ma'am, I'm unable to share the screen, ma'am.
Okay, we'll take next participant, Miss Ashita. Akshita. Miss Akshita. Akshita. Next participant is Mr. Sumit Joshi.
Yes, ma'am. Ma'am, am I audible?
Yes, you're audible.
Yes, ma'am. अरे यार कैसे दिखाऊं ये? Ma'am, screen is visible.
No.
अरे यार मैम
Yes, it is visible.
ओके मैम
Present your poster.
Yes, ma'am. Good afternoon, sir. Myself, Sumit Joshi from Smriti College of Pharmaceutical Education, Indore. Sir, my topic is liposomes as a gene delivery carrier. Sir, gene therapy is an active field that has progressed rapidly into clinical trials in a relatively short time. This therapy came in work for a few decades. Sir, gene therapy is a technique of genetic engineering to replace a faulty gene or a non-working gene to a normal functioning gene with the help of a vector, which is liposomes. Liposomes are generally a vesicles in which aqueous volume is enclosed by a membrane composed of phospholipids. The ability of liposomes to encapsulate hydrophilic, lipophilic, amphiphilic drugs have allowed these vesicles to become very useful drug delivery. Said in this gene delivery, the process of introducing foreign genetic material into the host cell by the help of vectors. Mm, liposomes in a gene delivery. Sir, the approach of a gene delivery, gene therapy, is the is to deliver the genetic material in the direct host cell by the help of liposomes for its proper functioning. The gene delivery must reach to the genome of the host cell to induce a gene for the proper functioning. The liposomes are often used to deliver a molecular cargo or a genetic material such as DNA for the therapeutic benefits. Hello?
Yes.
Yes, ma'am. Ma'am, the liposomes are the bilayer structures spontaneously formed by the hydrated phospholipids that are widely used as an efficient delivery system for drug as well as antigens due to their capability of encapsulate bioactive materials, hydrophilic type of drug molecules into inner water phase and within the lipids. It is having an improved understanding of thermodynamics, kinetics, and stability of complex for a better delivery vehicles for transfection will be constructed or happen with the liposomes. Thank you,
Sumit,
ma'am.
your time limit. Okay.
Yes, ma'am. Ma'am. Yes, ma'am. Ma'am, I am concluded
Yes.
myself. The liposomes, the key success for any gene therapy is a vectorable system for safe and efficient gene delivery by the help of liposomes. Thank you, ma'am.
Thank you, Sumit. Our next participant is Miss Yogeshwari. Yogeshwari.
Hello.
Yes. The next participant is Mr. Vaibhav. Vaibhav, unmute yourself. Vaibhav, you're not audible.
Good afternoon, ma'am.
Yes, you can present your screen.
Yes. Good afternoon, everyone. My today's topic is artificial
Vaibhav, we
intelligence.
can't see your screen. Firstly, present your screen. Are you able to share?
Yes, ma'am.
No, it's not visible. Vaibhav, are you able?
Yes,
Or will
ma'am.
take? No, we are not able to see your screen. Otherwise, we'll take next participant. The next participant is Miss Ritu Kumari.
Am I audible, ma'am?
Yes, you're audible. Ritu, are you able to present your screen?
Yes, ma'am. I'm trying, ma'am.
Okay, we'll take next participant, Miss Megha Gupta.
Yes, ma'am. Ma'am, it is visible?
Yes, it is visible.
Good afternoon to all respected teachers and my co-participants. I am Megha Gupta from Pharmaceutics, Indore Institute of Pharmacy. And I'm going to present on the topic nanosponge and its applications. The novel drug development and technology has brought a term targeted drug delivery. Targeting molecule to a specific site has brought a certain pharmacological action. Nanosponge is a part of nanotechnology, which is both hydrophilic and lipophilic in nature. They are colloidal carriers which improve the aqueous solubility, permeability, bioavailability in the body tissue. Nanosponge are tiny sponge which are in a size about virus less than 1 micrometer and it have cavity in their structure. In that cavity, we can load a large amount of the drug. There are various methods of preparation of nanosponge like ultrasonication assisted synthesis, solvent-based method, melt-based method, microwave-assisted method. The mechanism of release of nanosponge, here you can see in the figure nanosponge particles. It can be in the form of encapsulated based form. It can be in the lotion, cream, or injection. It applied to the skin and get penetrated through the minute pores into the cavity of the skin and reach to the blood carrier and bind to the cellular compartment, thereby reaching the desired targeting cell by giving the almost pharmacological action. There are numerous advantages of nanosponge like targeting site-specific delivery, provide improved elegance, stability in formulation, flexibility. It can stable into the 130 degrees Celsius and nanosponge have versatile compatibility with most of the vehicles and excipient. This technology provides better entrapment of active ingredient and has less side effect. It can improve the aqueous solubility of poorly water-soluble drug molecule. And it is an encapsulated type of nanosponge which protect the drug from the degradation. This technology is biodegradable and predictable release. There are various applications of nanosponge like molecular drug targeting as a toxin therapy, solubility enhancement as dermal penetration as an anti-cancer therapy. Nanosponge are the drug delivery system which can carry both hydrophilic and lipophilic drug. So we can formulate the different type of formulations such as oral, parenteral, topical, and this nanotechnology is application in the wide application in the pharmaceutical industries, and drug developed by this technology provide prolonged and controlled release of the drug in a safe and effective way. That's all. Thank you, ma'am.
Any marketed formulation available in market?
Yes, sir.
Can you name please?
Sir, for the tablet, it is glycine, which you have dexamethasone. And for topical solution, Mina Gargle, trade name, which have iodine. And for injection, it has paracetamol and capsule Braxin.
Thank you. Thank you.
Thank you, sir.
Thank you, Omega. Next participant is Miss Tanya Srivastav. Tanya, unmute yourself.
I did. Hello?
Yes, you're audible.
Yes, ma'am. So, good afternoon, everyone. Today, Tanya Srivastav, I'm going to present. Ma'am, is my screen visible? Hello?
Yes, it is visible. You can continue.
Okay, ma'am. Good afternoon, everyone. Today, Tanya Srivastav, I'm going to present my topic, that is liposome, a drug delivery system. But before that, I would like to thank Ms. Neha Kamalpuria ma'am and Dr. Dinesh Kumar Mishra for the help and guidance in completion of this presentation. Now, I would like to go straight to the topic without me wasting much of your time. Liposomes. The word is derived from the Greek word lipos, which means fat, and some means body. So they are fat bodies. Now you might think where liposomes are used, why they are needed. They are basically like spherical vesicles made of phospholipid bilayer containing drug or active pharmaceutical ingredient inside them, and they are used as drug carriers in drug delivery system. Now, why they are needed? If we introduce the active pharmaceutical ingredient directly inside the body, what will happen? It have the maximum chances of getting degraded before reaching the site of action due to the metabolism by the enzymes present inside the body and will show no therapeutic effect. So to overcome this problem, there is a need of carrier system. Now let us talk about its advantages. Because of its structure, that is phospholipid bilayer vesicles, they are suitable for the delivery of hydrophilic, hydrophobic, and also the amphipathic drug molecules. They are biocompatible, that means they do not show any harmful effect on the body as they resemble the biological membrane in form of spherical shell. They are non-toxic, they are completely biodegradable, they protect the drug from external environment, reduces the exposure of sensitive tissue to the toxic drugs. They increases the bioavailability, that means the amount of drug available at the site of action. They increases the therapeutic effect, the therapeutic level of the drug, that means the range of time in which the drug is expected to be effective. It helps to increase that. There are some disadvantages too, like the production cost is high. There might be a chance of leakage and fusion of encapsulated drug. It has short half-life. The stability depends on both phospholipid by which they are made and also on the active pharmaceutical ingredient which is encapsulated in them. The method of preparation, there are various methods to prepare liposomes. Some are thin film hydration, also known as Bentham method. The next is reverse phase evaporation, etc. The application Now let us discuss about the application of liposomes. They are used widely as drug carriers. For example, in
Tanya, your time limit is over. Please conclude.
Okay, ma'am. In conclusion, I would like to say that liposomes have been realized as an extremely useful carrier system for targeted drug delivery, and they are promising. They have promising future and will undergo further development for sure. So that's all I have to say. Thank you very much. It's done, ma'am.
Thank you, Tanya. Our next participant is Miss Momita. Tanya, stop sharing. Momita, please unmute yourself. Next participant is Miss Prachi Gupta. Prachi Gupta. Next is Anjali Arun Jaiswal.
Yes, ma'am.
Yes.
Is it visible, ma'am?
Yes, it is visible.
Okay. Good afternoon, one and all present here. I am Anjali Jaiswal, and today I am here to present you an overview on magnetically modulated drug delivery system. As we all know that drug targeting is the delivery of the drug to receptors or organs or any specific part of the body to which one wishes to deliver exclusively. So the magnetic drug targeting means the specific delivery of therapeutic agents to their desired target area using magnetic field. For example, tumors by using magnetic nanoparticles. Here we are using ferrofluids bound to these agents and an external magnetic field which is focused on the tumors. Well, these magnetic fields are believed to be harmless to biological system and adaptable to any part of the body system. And these magnetic particles composed of magnetic like rock minerals and one of the main iron ores and nickel, iron, cobalt and iron, boron, etc. and are all well tolerated by body. The principle of magnetic targeting is to in this technique, the drug is bound to magnetic compound and then injected into a patient's bloodstream and localized with a powerful magnetic field in the targeted area, as you can see in the image. And after that, depending upon the type of drug, it is then slowly released from the magnetic carriers, and a very high concentration of chemotherapeutic or radiological agents can be achieved near the target site without any toxic effect surrounding tissue or to the whole body. Well, these techniques give us some major advantages that target the specific area and reduce the system's distribution of drug and released in effective treatment at lower doses. The advantage of this technique is harmless to biological system and adaptable to any part of the body, and avoidance of acute drug toxicity directed against endothelium and normal parenchymal cells, and also adaptable to any part of body. Now the disadvantage is to magnetic targeting is an expensive technical approach and requires specialized manufacturing and needs trained personnel to perform procedures. The mechanism of magnetic targeting is to, in this technique, the drug transport technique is based on the fact that the drug can be either encapsulated into a magnetic microsphere or conjugated on the surface to the micro nanosphere. And when the magnetic carrier is intravenously administered, the accumulation take place within area to which the magnetic field is applied, and after that an external permanent magnetic field is applied on desired area to guide and concentrate the drug. Accumulation of the carrier at target site allow them to deliver the drug slowly. Well, this magnetic field form incorporated materials such as magnetite and iron, and these are normally grouped according to size at the lower end. And then we have ferrofluids, which are colloidal iron oxide solution. Well, then it encapsulated magnetite particles in the range of 10 to 500 nanometer, and the magnetic particles are just below 1 to 100 nanometer. This technique is used as a delivery of drugs. Now, the application of the drug is therapy of disease, targeting of radioactivity.
Anjali, your time limit is over. Please conclude.
Okay, ma'am. Now I would like to conclude this presentation. In magnetic vesicular system have been realized as extremely useful carrier system in various scientific domains. Over the years, magnetic microcarriers have been investigated for targeting drug delivery, especially magnetic target chemotherapy, due to their better targeting and tumor targeting, therapeutic effective, and lower toxicity and flexibility. Well, this is all about this topic. Thank you so much, ma'am.
Thank you, Anjali. Our next
Thank
participant
you, ma'am.
is Ms. Prachi Gupta. Prachi, please unmute yourself. Yes.
I was filled the form for oral presentation. Hello. Hello, ma'am.
Yes, Prachi, your name was for poster.
Ma'am, but I was filled the form for oral presentation, ma'am. So, ma'am, I have a PPT, not a poster. So, can I present?
No, that is have been done yesterday. Today is poster presentation.
Okay, ma'am.
Okay. Next participant is Ms. Harshita.
Yes, ma'am. Hello.
Yes. Ranjit, please mute yourself. Harshita, yes. You
Yes,
can
ma'am.
present your screen.
Yes, ma'am. Ma'am, is my screen is visible?
No, it's not.
Yes, ma'am. 1 minute. Ma'am, is it visible?
Yes.
Okay. Good afternoon, each and everyone present over here, respected teachers and my co-participants. My name is Harshita Gauraha, pursuing M.Pharma from Indore Institute of Pharmacy, and my today's topic is microspheres, an excellent technique for novel drug delivery system. As we all know, the targeting of active drug molecule to specific body sites with a controlled and predominantly drug release from the drug delivery system have great impact on human health care. Microspheres or microparticles have gained greater attention due to their free-flowing powder characteristics and biodegradable nature, generally made up of proteins or synthetic polymer. And the particle size ranges from the 1 to 200 micrometer. My focus on today's topic is on marketed preparation of microspheres. For example, risperidone. Risperidone is a drug used to treat mood or mental disorders such as schizophrenia. Risperidone targets the serotonin 5-HT2 receptors, dopamine D2 receptors, and alpha-1 adrenergic receptors. Risperidone microsphere were prepared with a suspension evaporation method. Suspension evaporation technique uses an aqueous suspension medium consisting of gelatinous inorganic gel which act as a suspending agent. Here in the preparation of risperidone loaded microsphere, aqueous suspension containing aluminum hydroxide gel, inorganic gel was used. Risperidone is dissolved in dichloromethane and separately aluminum hydroxide gel was formed. The drug solution was dispersed into the aqueous suspension medium consisting of aluminum hydroxide inorganic gel. The dispersion was homogenized and then solvent evaporated by stirring, and then concentrated HCl was added to the suspension to dissolve the suspending medium as shown in the figure. So solidified microsphere were removed by the filtration and washed with the distilled water. So in this technique, microsphere risperidone microsphere was prepared. So final overall conclusion on this topic is among multiple unit dosage form, microsphere plays an important role in area of particulate type of drug delivery system due to better entrapment and a small size with good release of characteristics and have many more advantages over conventional dosage forms like provide prolonged and constant therapeutic effect and reduces the dosing frequency. Taste and odor of the unpleasant drug can be masked by this preparation. Thank you. Hello.
Thank you, Harshita. Our next participant is Mr. Raja. Raja. Next participant is Ms. Samriddhi. Yes, you're audible.
बंद करना ना आया.
Tanya, mute yourself.
Hello, ma'am.
Yes, Samriddhi.
My screen is visible?
No, it's not.
Now, ma'am?
No, it's not.
Okay.
Are you able or will move forward?
No, ma'am. It's a problem.
Okay, we'll take next participant. Next participant is Miss Pranali.
Ma'am, am I audible?
Yes, you're audible.
Ma'am, is my screen is visible?
Yes, you can continue. Yes, it is visible.
Good afternoon, all the dignitaries, faculty members, and all my fellow participants. I am Pranali from Masters of Pharmacy from Indore Institute of Pharmacy, presenting my e-poster on 3D printing of pharmaceuticals. Three-dimensional printing is the most revolutionary and powerful technique which allows 3D shape of solid object or shape that has 3 dimensions, length, width, and height. It is also known as additive manufacturing, meaning creating an object by building by layer, by one layer by layer at a time. And it allows less wastage as compared to subtractive manufacturing. It is a versatile tool of precise manufacturing of various devices. Also, it serves as a technology for developing novel dosage forms, tissues, and organ engineering, as well as disease modeling. In 2015, Aprecia Pharmaceuticals produce sputum levetiracetam. It is an anti-leptic drug which became the first FDA-approved 3D printed drug by the European country. Then what is a 3D printing process? How it works? 3D printing process, first of all, we design a model with the help of a computer-aided design. And then we can take any filament material according to our choice for the development of the process, which can be PLA, polylactic acid, any polymer, powder, talc, resin, any kind of polymers. And then the slicing of the object and then the printing. Slicing of the object and then dispense according to the choice of your geometrical shape or any kind of shape that you want. And then printing of that object. 3D printing, there are different 3D printing technologies are used like fused deposition modeling, selective laser sintering, inkjet printing, zip dose, stereolithography. Benefits, it increases solubility, faster trials, doses specific parameters, it provides a tailored dosage form. Then what are the challenges we faced in 3D printing? Cost of pre and post processing.
Pranali, your time limit is over. Please conclude.
Lastly, I would conclude on that since 3D printing technology is a growing, but however, all the benefits for patients and overall healthcare system provided by the implementation of this technique, printing makes the required a lot of research to establish the process and according to the customized product. And it also faces several challenges in printing of the fragile nature of the object, which required well-planned procedures, and especially with the procedure, which are the products which are complex in nature. But since 3D printing is a revolutionary technique, so it can reshape the world, and technology can improve way from conventional pharmacy practice to a new advanced technology. Thank you.
Thank you, Pranali. Our next participant is Mr. Jatin. Jatin, unmute yourself.
Yes, ma'am. Am I audible?
Yes, you're audible. Jatin, please share your screen.
Yes, ma'am. Ma'am, is the screen visible?
No, yes, it's visible.
Okay. So very good afternoon to all the members. I am Jatin Gurnani and I am pursuing B Pharma from the College Indore Institute of Pharmacy. And currently I am in third years. My topic of today e-conference is nanomedicine in cancer and market industry of nanomedicine. Starting with the nanotechnology topic, the branch of science and engineering devoted to designing, producing and using structured device and systems by manipulating atoms and molecules at nanoscale. Mathematically, nanometer can be defined as 1 nm is equals to the 10 to the power minus 1 meter, leading towards the introduction part of the nanomedicines, in which I have discussed about the cancer part. Basically, as per the WHO report, there have been around 10 million death concern in 2020, among which the most common disease was breast cancer and then skin cancer, followed by the colon and many others. Materials and methods used in the treatment of cancer. Many common methods which are usually used like surgery, chemotherapy, radiation and gene therapy. Although these methods are very traditional and are currently used today also. Surgery, basically in surgery we use we have to cut down the tumor part or the microtumor environment from the human body, which can be dangerous, also leading to the death of healthy cells. In chemotherapy, we use various chemical medicines like alkylating agents. In radiation, we use a special power beam of light, which is used to kill the cancerous cells, and later on the gene therapy. But apart from that, the topic is nanomedicine, how it can be useful in the treatment of cancer. Basically, nanomedicine has a various parts like liposomes or doxil or polymeric liposomes and PLGA, that is poly-DL lactic glycolic acid, which are used in the treatment of cancer. Now, looking at this picture, if we use traditional drugs and nanomedicines, the traditional drugs is overall used in the body, and finally 2% of the drug is used, or very small amount of drug is utilized by the body. But in case of nanoparticles, the size is less than 30 nm, 40 nm, and between 30 to 40 nm. Now, looking at the evaluation method of the nanomedicine in treatment of cancer, it can be of two types: drug delivery based and drug targeting. In drug delivery, we learn various parameters like improving therapeutic index, low side effects, high enhanced permeability retention effect, multiple drug delivery to the tumor. So basically, here the topic comes that enhanced permeability retention effect. Basically, on this point, we have to enhance that how can be nanomedicine used in the treatment of cancer. Enhanced permeability retention effect is basically done in the drug targeting. Drug targeting can be of two types: active and passive. In basically active, the nanomedicine direct penetrate into the cancerous cells, which are basically leaking. And in passive, there is a peptide bond with the cells, leading to the less permeable into the tumorous cells. Now, results and discussion. The result is to get better safety and efficacy while we work on a nanomedicines. Number of nanomedicine has been continuously increasing and giving successful clinical trials followed by the treatment of cancer. But another part is P glycoprotein, which has been a concern in the terms of nanomedicine in treatment of cancer, as P glycoprotein act as a security guard for the treatment of cancer. In normal uses of medicines, P glycoprotein.
Jatin, your time limit is over. Please conclude.
Yes, ma'am. So my conclusion based on the nanomedicine used in the cancer treatment, the nanomedicine has been a powerful weapon in the treatment of cancer. Studies are being done that how it can be more effective, that how can we encounter more useful or successful treatments. And along with that, in market industry, North America has been leading as a powerful employer of the nanomedicines because of its best R&D development and the best API input. Thank you, ma'am.
Thank you, Jatin.
Yes, ma'am.
Our next participant is Rukaiya. Ms. Rukaiya. Next participant is Yash Jaiswal. Please unmute yourself.
Hello, am I audible, ma'am? Hello.
Yes. The next participant is Satish Agrawal. Satish. Next is Ankita Awasthi. Next is Mansu Patel. Next participant is Ms. Shipra Mahel.
Hello, ma'am. I'm audible. Hello.
Yes.
This is Sunil, ma'am. Can I present, ma'am?
Okay, you can present.
Okay. The screen is visible, ma'am.
Yes, it is visible.
Okay, so my topic is exploring anti-cancer potential of steroid alkaloid camptothecin in the treatment of cancer. So just as we know, the cancer is a leading cause of the death in the world, and the treatment available for the cancer of treatment is surgery, chemotherapy, and various natural therapies. So it has disadvantages. So we will move forward for the using herbal medicines for the treatment of cancer, and we are isolating camptothecin for the treatment of cancer isolated from nothopedes pneumonia. So we have isolated camptothecin from the nothopedes pneumonia and evaluated for the labdamax, LCMS, FTR, NMR, and evaluated for the DAPI and apoptosis by flow cytometry. So in that the FTR spectra of isolated camptothecin matches with then LCMS, the retention time will be 5.2.9 nanometer. That is exactly matches with the spectra, and animal spectra of the isolated camptothecin, which matches with the isolated spectra of the standard camptothecin. Then molecular weight will be 349.2, which is exactly matches, and we are evaluated this this isolated camptothecin for the treatment of anti-cancer IC50 value both prostate cancer and lung cancer. So we found that the IC50 value of camptothecin is less on the lung carcinoma as compared to the prostate cancer, and both camptothecin induces apoptosis and flow which is confirmed by further flow cytometry and DAPI evaluation. If you see the flow cytometry and DAPI images in the flow cytometry images, the camptothecin induces apoptosis in both lung cancer and prostate cancer, which is nearly about 23% of the cells in apoptosis stage, and also in case of lung cancer carcinoma, it is about 24% of the cells in apoptosis stage. So from the DAPI also we can say that it induces a nuclear webbing, nuclear morphology, and chromatin condensation, which shows in the DAPI images. If you clearly see chromatin condensation.
Thank you, Sunil. Our next participant is Miss Sapna.
सलोनी,
Yes,
आवाज आ
sir.
रही क्या मेरी?
Yes,
आवाज आ
sir.
रही है?
Yes, sir.
अच्छा,
You're audible.
ये जो कैंडिडेट है उसको 1 मिनट पूछना था मुझे.
Okay, sir.
सुनील,
Sunil, are you there?
है क्या?
No, sir. He's not here.
अच्छा,
He left
ठीक.
the meeting.
ओके, ठीक.
Okay. Our next participant is Miss Sapna.
Ma'am, am I audible?
Yes, you're audible.
My screen is visible.
Yes, it is visible.
So good afternoon to all. I am Shikna Saini, student of M.Pharm Pharmaceutics from Indore Institute of Pharmacy. Today my topic of poster representation is gene therapy, which is a current treatment approach. Gene therapy is a medicinal approach that treats or prevents a disease by correlating to a genetical problem. Basically, gene therapy is a recent approach which we are used in various diseases like cancer, cystic fibrosis, diabetes, heart disease, etc. It can use to be correct or replace the defective gene responsible. Basically, advantage and disadvantages of gene therapy are the basically main advantages promising a medicinal outcome, and the disadvantage is costly treatment because of the mutation of gene, and the study of gene is very costly. So it is the main disadvantage of a gene therapy. It includes basically 5 steps. First, creating a working gene. Second, building a therapeutic vector. Then third, determining eligibility. Fourth, delivering the working gene. Fifth, monitoring safety and efficacy. Basically, gene therapy divided into 2 types. First is DNA based and second one is RNA based. And now at the end, I want to conclude the whole presentation. Gene therapy is basically, sorry. Gene therapy is a potential to cure numerous genetic diseases. Like we can cure a number of diseases with the help of gene therapy, and which is a recent approach. Thank you.
Thank you, Samriddhi. Thank you, Sapna.
Yes, ma'am.
Our next
Thank you, ma'am.
participant is Vaibhav.
Am I audible, ma'am?
Yes, you are audible.
Okay. Screen is visible, ma'am?
Yes, it's visible.
I am Vaibhav Devare from Smriti College of Pharmaceuticals. Today's topic is artificial intelligence in pharmaceutical industries. Artificial intelligence means the way the computer can be made to copy the human thing. Artificial intelligence has made revolutionaries from in recent years for the drug development processes. Artificial intelligence produce the physical data, but there are some challenges to which a great powerful tool in which information in the field of drug design. Partnership between artificial intelligence and pharmaceuticals companies. The data which is gained from the industries on the basis of fragment screening, computational modeling, induction and deduction processes alternates, which eventually leads to hit and lead compounds. Or the de novo drug design is another technique in which it requires the knowledge of the organic chemistry for in silico compounds and its synthesis for the development. The next technique is the virtual screening model that functions as surrogates for biochemical and biological tests of efficiency and toxicity. Means that active learning, it requires expertise in algorithm for the development of the new and novel compounds. In this, the first step comes that it requires the identification of the novel chemical compounds for the enzymes or biological activity. And the second step is a chemical compound that contains the lead, show the promising potential that can lead to the development of the new drug for the treatment of the diseases. The next is the QSAR. It means a quantitative structure activity relationship. It means there is a relationship between the chemical structures within the activity using the statistical approach. QSAR helps to know the untested chemicals results. It is a method which developed during the past decade. Most of them generally focuses on the drug discovery.
QSAR model can be used for the identification of the chemical
Vaibhav,
compounds.
your time limit is over. Please conclude.
Yes, ma'am. Conclusion is that we have seen advancement in recent years, and over next 2 to 3 years, experts are very sure that there will be a change in the pharmaceutical industries, the way of the drug discovery. Using this technology, individual, the one who is performing, should be trained and requires a domain expertise. Thank you, ma'am.
Thank you, Vaibhav. Our next participant is Advita.
Yes, ma'am.
Sorry to interrupt, Ms. Saloni. May I know how many presentations are left?
So 3 more.
Okay.
Ma'am, is my screen visible?
Yes, it is visible.
Good afternoon, everyone. My name is Advita Pandav. I am student of M.Pharma first year from college, Smriti College of Pharmaceutical Education. And my today's topic for poster presentation is anti-cancer activities of natural compounds. Cancer is an abnormal growth of cells in our body, which leads to death. It basically destroys the normal cells, which creates imbalance in the body. The majority of the anti-cancer drugs causes toxicity and are very costly and it shows side effects. The causes of the standard techniques for treatment of cancers are radiotherapy, chemotherapy, and surgery. In current situations, that traditional methods of treating cancer are being used. Many herbs and natural compounds are being used, like tea, neem, haldi, sadawar flowers, and turmeric. Among these, turmeric is used very often. Curcumin, which is extracted from turmeric, is widely used as it suppresses the activation of nuclear factor kappa B, which is a protein transcription factor, which leads to suppress many gene involved in tumor genesis. Curcumin is also involved in cell cycle control and stimulation of apoptosis via regulation of protein P16 and P53. Curcumin is the conclusion is using of natural compounds in treatment of cancer can lead we can have a better treatment for cancer, and it will be less toxic to our body, and which will show less side effects. Thank you.
Thank you, Advita. Our next participant is Ms. Samriddhi. Next is Ms. Rukaiya. Next is Ms. Ritu.
Am I audible, ma'am?
Yes, you are audible. Please present your screen.
Yes, ma'am. Visible, ma'am?
No, it's not.
Screen.
No, it's not. Are you able to present?
Go for next
Yes,
if
ma'am.
there is a technical problem.
Yes.
It's visible, ma'am?
Yes. Poster is not visible.
I request all of you, please prepare in advance because
Ma'am,
please
poster is
follow
visible.
the timeline.
No, it's not. Okay, we'll take next participant. Miss Mansi. Miss Mansi Patel.
Please proceed for further. Next participant.
Also, this was the last. Okay, so I would like to thank all the participants for their presentation. And I would like to thank you, Dr. G.N. Dharwekar sir and Preetam Siraskar sir for joining and giving a valuable time. Now, I would request Principal sir to please present certificate of appreciation.
Thank you, sir.
Principal sir, Dr. Dinesh sir. I would request Principal sir to please present certificate of appreciation.
Thank you, Saloni. Thank you, Dharwekar sir, for
Thank
your
you, sir. Thank you.
for your gracious presence and sparing your valuable time. I know that today I called you many times and you were busy in the meeting, but in spite of your busy schedule, you spare your valuable time. So thank you very much on behalf of entire organizing team and Indore Institute of Pharmacy. I would like to give the certificate of appreciation. So sir, please accept this certificate. And once again, thank you very much, sir.
It's being my pleasure to be here to listen the views of the upcoming pharmacist.
Right, sir.
Yeah. Some of the topics were really good. They have chosen a very good topics.
Right.
Though initially there was some glitch on the audio, so finally I joined with my mobile and then I could communicate. Okay.
Right.
Otherwise, I was there from the first candidate to the last candidate.
Right, sir.
Thank
Sir,
you.
Thank you. Yeah, I also would like to thank Mr. Preetam R. Siraskar, Production Manager, Glenmark Pharmaceuticals, who also spared his valuable time because he was in company, but since I requested him, so he came and he spared his valuable time with Dharwekar sir. I think he also enjoyed the session. So thank you very much, Preetam sir.
Thank you, sir, for giving me this opportunity. And I really want to appreciate the participants because the topics which they have chosen is really more interesting, like artificial intelligence, QBD, and there's Six Sigma. So I must say the students are in a very right place and in right hand. So it will be a benefit for further in future from quality pharmacists will become.
Okay.
Thank you, sir.
Thank you, Darvikar sir. Thank you, Pritam sir. Thank you, Saloni.
Thank you.
Thank you all for joining. Now we'll have a short break and we'll restart a valedictory program at 3. Thank you for joining. Please stay connected.